Well, good morning, everybody, and thank you so much for joining us this morning. My name is Tim Illidge, and I'll be chairing the meeting on behalf of the Manchester RadNet. This is the first of the RadNet Radiotherapy Immunotherapy meetings, and it's a real pleasure for us to be hosting it. I'm really, really grateful for the quality of the speakers that we've got today, that they've been willing to give up their time to participate in this meeting. If I could ask all the participants to keep their microphones on mute and the videos off to save the bandwidth. Any questions that you have, could you please put them in the chat addressed to everyone. The agenda is also in the chat, so you can see who's coming up and what we're doing. So it is interesting that as we enter the 21st century, we've moved away from focusing entirely on almost entirely on last century on how radiotherapy affects the tumour cells, to become interested in how the immunomodulatory effects that radiotherapy might have. And in particular, the excitement of this has been the ability of radiotherapy to induce immune responses, both local and systemic immune response. The challenge, however, is that these abscopal effects, the so-called immune responses that you get systemically, are extremely rare. In fact, when we looked carefully at this, there were 23 cases that were reported over the last 54 years, which makes these abscopal effects actually rarer than the sightings of the Loch Ness Monster over these last 54 years. So although this is extremely exciting, it's in the clinic is a very rare phenomenon. So in an attempt to increase the number of immune responses, the combinations with immune checkpoint inhibitors have been really rather interesting and important. But again, these are extremely rare events. So if we look at this particular abscopal effect in a patient with melanoma that had an anti-CTLA-4 antibody, the numbers are quite alarming. That although this is great for the authors with 1,500 citations and 22 authors, there was just one patient. It would be great if we had 15,000 citations on 1,500 patients, but that isn't the case at the moment. So why is it so difficult to generate meaningful immune responses with radiotherapy? Well, that forms the focus really of what we're going to be talking about this morning in terms of the immunosuppressive tumour microenvironment shown in this cartoon here. A number of the key cytokines and immune effector cells, including the T regulatory cells that we'll be focusing on with Sergio later, the myeloid-derived suppressor cells, these tumour-associated macrophages. And this morning's presentations are really about ways that we might overcome this immunosuppressive microenvironment. So I'm extremely grateful for the speakers and the quality of the speakers that we've got this morning to talk about some of the challenges that we have if we're going to make progress in really generating meaningful systemic anti-tumour immunity. So I will talk initially and give an idea of the breadth of work that's going on in Manchester. Sergio Quezada will talk about novel T-reg targeting antibodies. Alan Melcher will then follow, looking at immune gene expression patterns during neoadjuvant radiotherapy in rectal cancer. Amato, Giaccia will then talk about studying immunological resistant tumour clones. Can that lead to more effective radiotherapy? Then we'll have a 15-minute comfort break, and after that comfort break, Kevin Harrington will talk to us about combining DNA damage response modifiers, Adel Samson about oncolytic viral therapy, and Awen Gallimore will complete what I hope will be a very exciting morning, talking about novel approaches for optimizing the tumour-specific T-cell priming. And after that, we will have a discussion about what we've been talking about this morning, and hopefully the way forward as to how we might form a useful networking group. So my presentation is aimed at providing the breadth of what's going on in Manchester without going too deep into any of a number of stories. I'm hoping that in subsequent meetings we might pick up some of these individual stories and talk about them in greater depth. And I've deliberately wanted to go show you the breadth in order to hopefully provide some interest and to stimulate some conversations and hopefully collaborations across the RadNet network and beyond. When we think about the radiotherapy therapeutic ratio, I think for too long, like in the last century, we've been focused entirely on the tumour cells and ability to sensitize these tumour cells with chemotherapy, which overall, after many decades of effort, has really made very little progress in adding to human happiness, with the exception of one or two solid cancers. Generally, the problem with adding chemotherapy is we haven't increased the therapeutic ratio, but have simply increased the toxicity that we've seen. There have been many attempts with oxygen and hypoxic cell sensitizers, which have had mixed results. But the major point in thinking this through is that we haven't really given a lot of credence to the tumour microenvironment and the immune effector cells. And if, for example, we were to hypothesize that the radiotherapy-induced immune dictators dictate both the tumour control and the normal tissue inflammatory response, if that is important, then we really require new models for thinking about enhancing the therapeutic ratio that include the tumour microenvironment and the radiotherapy-induced modulatory effects. We've known for some time that these immune effector cells are really very sensitive, some of them, to the effects of radiotherapy, but we give little thought with the modern techniques of VMAT and IMRT as to spraying the dose around as to how that affects the B cells and the T cells in the systemic circulation. And it's likely that that is probably very important to the generation of systemic immune responses. So there are a number of ways that we can potentially improve clinical outcomes combining radiotherapy with immuno-oncology agents. That from this point on, I will talk about them as being IO agents. The area which has derived most interest to date has been in the metastatic setting, where radiotherapy has been used to enhance the systemic immunity of an IO agent. And there, the goal is potentially converting oligometastatic disease from the incurable to the curable. But perhaps the most significant progress to date has been made in local disease, where the IO agents have been added to improve local control after chemoradiotherapy, for example. And a very good example there is the specific trial in non-small cell lung cancer, which has really improved outcomes for those patients with non-small cell lung cancer, albeit the majority still need better therapies to improve their outcomes. So we've been looking in the laboratory for some time as to how radiotherapy and immuno-oncology agents can generate systemic immune responses. And we found that in some tumor models, this model is the CD26 colorectal model, that radiotherapy in low doses, just 10 gray in 5 fractions, with anti-PD-1 or anti-PD-L1 leads to systemic anti-tumor immunity, and the scheduling of this was important. In other words, it was important to give the anti-PD-1 during the radiotherapy, and if we gave it after the radiotherapy, we lost the therapeutic effect. Now, in keeping with what we see in the clinic, this is actually, this generation of systemic immune response is only seen in very limited tumours. So we see it in the CD26, we see it in some melanoma models, but it's not seen in the prostate cancer models, where the addition of the anti-PD-L1 to radiotherapy makes no difference at all. And it's not seen in the breast tumour models here in the 41. So we've been focusing in the laboratory on 3 large areas, 3 significant projects. So when we do see this systemic immunity when radiotherapy is added to anti-PD-1, we've been looking at what are the mechanisms of that and the interplay between the tumor resident cells and those cells which actually infiltrate into the tumour following the treatment. So here we've been investigating the T-cell migration and their kinship. Another area of interest has been trying to convert these non-responsive tumours into responsive tumours, which is what many groups across the world are now doing. And I will talk more about this in a moment as to some of the IO agents that we've been interested in investigating. And the hypothesis here has been that these myeloid cells play an important part in this resistance, and if they can be therapeutically manipulated, we may be able to improve the tumour control. But aside from the immuno-oncology agents, what we think is also critical to success here is thinking more imaginatively about the delivery parameters of radiotherapy, in particular the dose and the fractionation, as well as the volume of tumour and or regional nodes that are treated. So we need to think really about the importance of the radiotherapy delivery, because I personally think it's most unlikely that we're going to have any or limited success using a single site of radiotherapy, and that we need to think in oligometastatic disease about irradiating more of the tumours, and in localized disease we need to think carefully and be informed in our decision making about the importance or not of irradiating the regional lymph nodes in terms of the volume of tissue that we irradiate. So we've been developing models that we hope will inform clinical trial design, rather than embarking on large and expensive clinical trials without the preclinical proof of principle to increase the chances of success. So in collaboration with colleagues in Belfast, initially in Mills Laboratory, we've been developing this DVL3 mouse prostate tumour, which lacks both P10 and P53 genes, common in metastatic and advanced prostate cancer. As a model of prostate cancer, we've also been developing orthotopic bladder tumours. Rick Walshaw has been leading this work for us in our lab, where we can use ultrasound to inject the tumours with TLR3 agonists in this case. And what's become clear is that we need more sophisticated and accurate ways of delivering image-guided radiotherapy to simulate what we're seeing in the clinic. So one of the observations we made some time ago is that in many of the tumours that we look at, irradiating these tumours actually increases the number of myeloid cells further. So if we look at, for example, at this prostate cancer model, the DVL3 model, we can see that the number of T cells in it is really very low indeed, and in contrast to the melanoma model. And when we irradiate the melanoma model, we see an increase in the number of CD8 T cells that go into the tumour. In stark contrast, in the murine prostate DVL3 model, when we irradiate the tumour, what we see is a large influx of these CD11 B cells, which are shown here in the bottom right, with these white, increased number of these white blobs, which are these CD11 positive cells. So what this is telling us is that the radiotherapy can make an immunosuppressive environment actually seemingly worse. So we've been looking at ways that we might potentially target and reprogram these tumour-associated macrophages and the myeloid cells. There are clearly a number of ways that we can potentially go about this immunosuppressive environment, shown here on this excellent slide from Jeffrey Pollard's review. And you will notice, you go around this rather busy slide, that many of these approaches have been and are being adopted in clinical trial and in preclinical investigation. To date, on the left-hand side of the slide, the depletion approaches using anti-CSF1 and anti-CCL2 have been unsuccessful, and we've had limited, or albeit extraordinary breakthrough success with the anti-PD1s in some tumours, but the majority of the patients are not responding to these therapies outside of malignant melanoma, where it's been an absolute game changer. So our approach has been to use the TLR agonists and the TFR receptors, OX40 and CD40, to think about ways of reprogramming these myeloid cells. So this is just a slide which shows some of the evidence we have, preliminary evidence for reprogramming of these monocytes from the M2 to the M1 phenotype with a CD40 ligand, where you can see that the M2 markers are shifted to the left and the M1 markers are shifted to the right. When we look at immunohistochemistry, we see that there is an influx of these CD8 T cells into the tumour. And importantly, when we add this ligand to radiotherapy, we see that there is an improvement in the tumour control. What we've also noticed is the scheduling of the anti-CD40 agents appears to be important in terms of the therapeutic outcomes. And in contrast to what we saw with the radiotherapy, the anti-PD-1 in the CD28 model, we need to give the radiotherapy prior to the anti-CD40 to see the maximum effect here. And it's when we give the CD40 later that we begin to see this improvement in the therapeutic outcome, and this is in a prostate cancer model. We've also been looking in lung cancer, and this is data from clinical research fellow Tiana, who's in collaboration with Carl Martins in Cambridge. We're using this KRAS P53 null adenocarcinoma model in lung cancer, KP475, as well as the squamous cell model, the KLN205. So this is some preliminary data which essentially confirms the radioresistance of this KRAS model. Essentially, 3 8-grade fractions have little or no effect over the non-treated controls. In contrast, when we use anti-PD-1 with anti-OX40, we get durable responses. That's not seen with the anti-PD-1 alone or the anti-CD40 alone, but the addition of this combination, anti-PD-1 to the OX40, to radiotherapy, doesn't lead to any more significant growth, long-term growth control. So Tiana's been looking and probing the mechanisms for this, and that's for another day to go into more details. But needless to say, common to a lot of these stories, it has been the generation and influx of CD8 T cells, which brings about the durable control. And when we deplete the CD8 T cells, we completely lose this therapeutic effect. Another agent that we've been interested in exploring is BO112, which is a TLR3 agonist. It's a double-strand RNA poly-IC, and it acts by activating the TLR3, RIG1, and MDA5, leading to an immunogenic cell death and increasing type 1 interferons, T cell activation, and immune checkpoint inhibition. So we see this, the interferon, the CD8 T cells, and we've been working to study the mechanisms in a number of different tumor types and translate this into clinical trials. Rick Walshaw, has a clinical research fellow in the lab, has been leading the work in orthotopic bladder cancer models. And what Rick has found is that the drug itself, BO112, induces immunophenotypic changes on the tumor cells, direct tumor cell death in vivo, activates DCs, T cell priming, and the induction of the interferon gamma pathway, and also potentially brings about the redifferentiation of macrophages to the M1 phenotype. And we've already started to translate this into the clinic with a clinical trial looking at the injection of BO112 into locally advanced bladder cell cancer models. So last year we were successful with colleagues in a CRUK Accelerator Award led by Nacho Malero and Plamploma, and here with colleagues in London with Sergio and Crispin, as well as immunologists in Italy, we'll be looking at local injection in combination with radiotherapy. So there are a number of different agents that we can inject to bring about, we hope, local priming with intratumoral injection and of immunostimulatory agents to trigger the tumor-specific immunity. Many of these, this pioneering work was done by speakers later on this conference. So Kevin Harrington and Alan Melcher have done really world-leading work with oncolytic viruses with local injection. We'll hear a little bit about that later on, and that work continues in Leeds, which we'll hear about later on the call. But this is an approach which we hope will be useful in dissecting some of the mechanisms and understanding what happens within the local environment and how we might better generate systemic immunity. So now switching from the laboratory to the clinic, the big question is, how do we make progress with clinical dynamic radiotherapy immune biomarkers? So what we know is that radiotherapy can cause immunomodulation in the tumor and microenvironment, but we really don't know whether that can be advantageous. That's to say that it shifts things to the left and brings T cells into the tumor, or may, in fact, make matters worse by bringing about the recruitment of more myeloid immunosuppressive cells. So what is clear is that owing to the complexity of the tumor immune interactions, that static biomarkers at the beginning of radiotherapy are unlikely to be sufficient to accurately predict the response. And what is required is the dynamic markers, so we can see in real time how the radiotherapy is affecting this microenvironment. Of course, that's not without its challenges in being able to get biopsies during or shortly after patients are having radiotherapy. But that challenge is what we're attempting to do in Manchester, and would love to do this as a collaborative effort across more of the RadNet and radiotherapy sites in the UK. So here what we're proposing to do is a pre-treatment biopsy where we look at the immune profiling. The patient has radiotherapy, and we look either during the treatment or immediately after the treatment, such as in rectal cancer or cervical cancer, as to how the radiotherapy has altered that microenvironment. We are hoping, as we get more information on this, that it will be informative in terms of how we might manipulate that microenvironment further with either immune checkpoint blockade or the use of immunostimulatory agonists for those types of tumors where they are T cell poor, they have low PD-L1 expression and high numbers of myeloid cells. So we'll be doing this dynamic immune tissue and blood biomarkers, looking both in the tumor for immune gene signatures, immune cell infiltrates with the multiplex immunohistochemistry, looking at the T cell repertoire diversity, and looking in the periphery for the plasma cytokines, the chemokines, and the T cell repertoire. The final area I wanted to touch on this morning from the work that's happening in Manchester is a new and exciting collaboration with the Lydia Becker Institute for Immunology, where there are world leaders in mucosal and barrier immunity. And it's been a real pleasure to interact with Professor Mark Travis and Dr. Doug Dyer, who are experts respectively in the regulation of barrier immunity and the cytokines that are critical for that. So one of the projects that we'll be looking at is how radiotherapy influences the regulation of the barrier immunity and the homeostasis by the integrin-mediated transforming growth factor TGF-beta activation. And Ursula Seiselak has been the link between the two groups as we are going to start this work with Kay Williams. And this was going extremely well prior to the COVID-19 pandemic, and we're just beginning to restart this work, looking at how radiotherapy induces both acute and late normal tissue injury and the immune cell activation that leads to chronic inflammation. And we're hoping that if we can get more mechanistic insights into this, we'll be able to manipulate it and improve the therapeutic index. So to conclude, what lessons have we learned? from the laboratory. We know that radiotherapy leads to the T-cell infiltration in the irradiated tumour. In some tumours, that's probably the minority. The T-cells produce the interferon, which drives the PD-L1 upregulation on the tumour cells. We know that this is an adaptive mechanism in some tumours. We know that for these tumours, in this situation, when radiotherapy and anti-PD-L1 are combined, or anti-PD-1, this leads to the CD8 T-cell infiltration into the non-irradiated tumours, and that can increase the T-cell diversity and induce the abscopal responses. However, that only occurs in a small minority of tumours, we think, and in many common tumours like prostate cancer and breast cancer with low levels of CD8 T-cells, but high levels of myeloid cells, the use of immune checkpoint blockade in combination radiotherapy is likely to be largely ineffective in these tumours, in our view. So in these tumours that are resistant to immune checkpoint blockade, other approaches are needed. There are a number of promising immunostimulatory agents. Our focus has been on the TNF receptor family, where we're looking at anti-CD40 and anti-OX40, as well as the TLR3 agonists, which I mentioned briefly. Certainly in the clinic, we need further studies urgently to investigate the importance of radiotherapy dose, fractionation, and the volume that we're irradiating. And we need to look at the scheduling of the IO agent, which is likely to be very important. Maybe tumor specific and also maybe IO agent specific, but we need to give more time and thought into the scheduling of agents. We need to look particularly at the dynamic biomarkers of radiotherapy and begin to look in real time how radiotherapy alters that tumor microenvironment. If we do these things, and we have a great potential for radiotherapy and IO agent combinations to really improve outcomes for the many patients that we look after, given that around half of them are getting radiotherapy and their outcomes could be improved further with these sort of approaches. So my final slide is to thank members of the group, and particularly long-standing working relationship with Dr. Jamie Honeychurch and the Ellie Cheadle, who's been doing the clinical translation work, Divine Mukherjee, who was doing the prostate cancer work, and then the clinical research fellows. I briefly spoke about some of their work, Tiana and Rick. And thank you very much for your attention. And I shall try and stop sharing now and go to the questions.
So I have a quick question for you.
Yeah, go ahead, Sergio.
It's based primarily on the data you're showing, the combination. I think that's really cool. So there are two things. One is that there was no further addition, neither positive or negative, right? Which is a good thing, of the radio to the OX40 PD-L1 combinations.
Yeah.
If I remember that.
The lung cancer models.
In your lung cancer models.
Yeah.
Are those Treg driven models? Are they heavy on Tregs? And when you take the Tregs, then basically you just unleash the brakes, and that's what the OX40 is doing. So you don't see any further addition on top of that? Yeah.
So that's a great question, Sergio, and one that we haven't specifically addressed. What we see is that the number of Tregs is depleted with the OX40 and the anti-PD-1 combination. I rather race through that, but there is a significant reduction in that Treg population.
Yeah, that's expected because you're probably using the OX86, which is a depleting isotype. Although people don't talk about that, but that's the isotype, right? Yeah. But if the tumor is then Treg heavy and Treg dependent, then the data is great, but you're just not giving yourself a chance to look at the impact of the radio on top of that. Right, so you need a model that is less Treg dependent in a way. I don't know if that makes sense.
Yeah.
Because it would be really good to see the potential synergy there. Maybe using a non-Treg depleting anti-OX40, one that is a pure agonist.
Well, that's definitely worth a longer conversation, Sergio.
Yeah, sure.
So we've got some questions coming in. Does the number of fractions of radiotherapy make a difference in your opinion? So the preclinical data for this are, I would say, somewhat controversial and mixed, and we really haven't got good clinical data. My view is that it needs to be explored, and it probably does make a big difference, but that difference is probably going to be tumor dependent for the number of fractions. So I think that is an area that we need to study. Another question is there any additional toxicity when combining with the anti-CD40 antibody? And the answer is, in the mouse models, no. But I think that needs to be explored on the clinical translation, and it is likely that there will be additional toxicity, but it needs to undergo a rigorous clinical testing there. We know for the single agent anti-CD40 that they are tolerable. Efficacy was not as high as hoped for, but they were certainly manageable. So I think it is definitely feasible to test the radiotherapy anti-CD40. I think in the interest of time, we need to move on. We have got some time later on to pick up some more questions, but if I could hand over to you, Sergio, to share your screen and give your presentation, that would be great.
Sure, thank you. Thank you, Tim, for the invitation. It's really a pleasure to be part of this amazing lineup. So I'm going to be discussing some work in my lab related to the development of new antibodies for Treg depletion in mice and humans. This story really starts quite a long time ago when I was working in Jim's lab, trying to understand how his anti-CTLA-4 antibody work. We're looking at imaging in mouse models of cancer. Basically, what we saw is that in poorly infiltrated melanoma models, this is kind of the picture you see. You have a paucity of CD8 cells, and then you have patches of infiltration by regulatory T cells. So poorly infiltrated tumor dominated by Tregs. When you treat this animal with a vaccine and only with the vaccine and the anti-CTLA-4 on top of it, we saw a very significant change in the landscape with an influx of CD8 cytotoxic cells and what appeared to be a reduction in Tregs. But we thought that it was a fraction issue. So you have more effectors, hence it looked like you have less Tregs. But long story short, we were wrong, and really what we demonstrated in 2013 and in a series of papers following up that is that anti-CTLA-4 antibodies are actually, in mouse models of cancer, are actually depleting antibodies, and people did not know that. And that for an anti-CTLA, when an anti-CTLA-4 antibody in mice gives you a response, it's because it's depleting Tregs primarily. So we catalog them as dual activity antibodies. The mechanistic reasoning is the following. The Treg inside the tumor express many more molecules of CTLA-4 on the surface. The effector cell does express CTLA-4, but few molecules on the surface. So when you have a lot of antigen on the surface of a cell, and then a good antibody with the right isotype, which happened to be the one of the anti-CTLA-4, then that binds to Fc receptors in macrophages and NK cells, and that triggers what's known as ADCC, or antibody-dependent cytotoxicity. So that's the first activity of the anti-CTLA-4. The second activity continues to be the checkpoint blocking activity, right, where it blocks the binding of CTLA-4 to B7 and then release the brakes on effector cells, and that gives you a second loop of activation. So that's basically what we demonstrated, supported by many others. There's no real argument about this activity in mouse models of cancer, and this really triggered not only academic, but also company-wise work in trying to develop a next generation of anti-CTLA-4 antibodies with enhanced ADCC for use in the clinic, right? And we were part of that wave, quite interested for a while, until we start looking at this data. So this is human tumor-infiltrating lymphocytes from patients with melanoma. And what we're looking here is CTLA-4 on the surface, on the y-axis, and ICOS on the x-axis. CTLA-4 positive, ICOS positive T cell, either a Treg or an effector, is an activated T cell by definition. And when you look in the tumor, you see what we have described before, that the Tregs express a lot of CTLA-4 and ICOS, are very activated. The helper cells, a fraction of them do express it, and the CD8 cells, which are the cytotoxic, they express much less. Remember, this is human melanoma cells. So if you were to put an imaginary line, which is how antibodies work, right? They need a high density of target. Most probably what the current anti-CTLA-4 antibodies in the clinic are doing are having some impact on the Tregs, some potential impact on this population of CD4s, which we don't know what it is, but they're really not touching the CD8 in terms of depletion, right? Which is good, that's critical. The concern that we had is that if we start increasing the affinity to Fc receptors and making a better depleting antibody, then that threshold, it's gonna go down. and you enter a remedy in which you might end up doing some damage by killing activated effector cells, right? So we didn't like that. So we decided to work on developing CTLA-4 antibodies with enhanced ADCC, and then move into a target, or try to find a target that was cleaner. And the target that we've been working in parallel for many years, since my PhD actually, CD25. So a little bit of new biological insight into this molecule. So CD25 is really the high affinity receptor for interleukin-2, and interleukin-2 is a critical growth factor for T cells. It signals via STAT5 and then another set of cascades illustrated here. But it's a quite a complex cytokine because its activity is different in vitro from in vivo. But bottom line, it's a cytokine that is critical for T effector activation, also T regulatory cell activation, and NK activation. So it's a complex cytokine, very important. It's highly expressed, the receptor, it's highly expressed on Tregs, right? Which makes you think, oh, this will be a great target to kill Tregs. But it's also upregulated on activated effectors in vitro. So if you take any effector T cell, you tickle them through their T cell receptor, they will upregulate CD25, which makes you think, well, I don't want a depleting antibody because I'm going to kill my effector cells. But careful, that is in vitro. If you look at the expression profile in mouse tumor infiltrating lymphocytes, these are 3 different tumor types. Just focus on the last one, which is the most immunogenic one. The data is the same across. And you look at CD25 expression on the surface of the different T cell subsets infiltrating a mouse tumor, you see that CD25 is primarily restricted to the FOXP3 Tregs. It's expressed only in a tiny fraction of CD8 cells and at low levels, and it's expressed in a fraction of the helper cells. So in my book, this looks like a very nice target in mouse models. Like a depleting antibody, it's likely to take these guys down and spare the vast majority of the CD8 cytotoxic cells. That's in mouse. What happens in humans? It's pretty much very similar data. This is human melanoma, metastatic, and treatment-naive non-small cell lung cancer and renal cell carcinoma, tumor patients and blood patients. That's incorrect. It's not lymph nodes, it's blood. So when you look in tumors, CD25 is preferentially restricted to the Tregs, the FOX positive, and not expressed by the CD8 cells infiltrating these tumor types. So again, looks like a pretty decent and specific target. That's primarily treatment naive. So what happens in the concept of immunotherapy? This is a collaboration with the team at the Royal Marsden, with Samra Turaklik on a clinical trial in renal cell carcinoma, where we can access a patient's tumor biopsy at baseline and a second biopsy on the same site on treatment. And this patient is being treated with nivolumab, right? So in the context of anti-PD-1, we can look at the expression of FOXP3 and CD25 on, the expression of CD25 on the effector compartment and on the regulatory compartment. Can you guys see my mouse moving, right? I hope.
Yes.
Yes, we can. Yes.
Good. Thank you. So basically what you see here is at baseline, the brown CD25 is preferentially associated to the FOXP3 in blue and not to the red cells. And the same thing is maintained even if this patient has been treated with anti-PD-1. So even in the concept of immunotherapy remains a good target. And this is the quantification done blind by Teresa Marafioti. So stays like that. And we tested this in a second trial called ADAPTEM, which is also melanoma patients treated with anti-PD-1. So if this looks such a juicy and nice target, why was it dropped? It was dropped a long time ago. It was dropped because of this type of experiment. When the anti-CD25 antibody came out and people started using it in mouse models of cancers, we all use a clone called PC61, which is a rat IgG1. And if you inject it into a mouse and then you look in blood, spleen, and lymph node, the Tregs are gone in 4 days. So it's a good depleter, right? In that context. So the experiment that we all will do, it was the one that you have a suboptimal therapy. Let's say in my case was Gvax and anti-CTLA-4. At day 8, it doesn't do anything. That would be the black line. It doesn't protect your mice. And you combine that with a prophylactic depletion of Tregs. So you deplete your Tregs with the PC61, wait 4 days, you check in the blood of the mouse that the Tregs are gone, they're gone, and then you challenge with the tumor, and then you give your suboptimal therapy. And that takes you from the black line to the blue line, and everybody's happy. Oh, great target, blah, blah. But the real experiment is a therapeutic intervention. It's not the blue line, but it's the red line. So you challenge the mouse with a tumor, and then you therapeutically deplete the Tregs, and then you treat with your suboptimal vaccine, and that never worked. We published that, and many others published that, and the argument was always that in the tumor, you have activated effector cells that upregulate CD25. and then your antibody is killing them. That was always the argument. But I think I show you plenty of data to suggest or demonstrate that CD25 is not upregulated in activated effector cells in the tumor. So what was going on? And this is what Fred Archie in my lab demonstrated, is that if you have an untreated animal, you have lots of Tregs in the tumor, FOXP3 and CD25. But if you give the classical anti-CD25 that we were all doing, and you look in the tumor, you see, oh, the CD25 is gone, but actually the FOXP3 cells are not gone. So the antibody is not depleting activated effector cells. It's actually failing to deplete anything. It's simply inducing a downregulation of the CD25 target. So it's giving you false impression of depletion. And that's because the antibody, because of the isotype, that particular isotype, it's a really bad binder to the Fc receptor required for ADCC in mice, right? So the fault is ours and all of the field for being using the wrong isotype or for not understanding isotype behavior, rad isotype behavior in mouse models. Now you can fix that by engineering the antibody and converting it into a classical mouse IgG2A, which is now a good depleter. So the antibody will bind to the same epitope on CD25, but now engage with Fc receptors. And when you do that, you get to very nice 44% of Tregs in the untreated, and with the new antibody, you get a nice fourfold reduction. So you can fix the depleting activity of the antibody by doing some simple engineering. So now that we fix the depleting activity, now we have a decent target to synergize with anti-PD-1 therapy or to give you single agent activity. So we did the experiment that we have failed before, which is to challenge the mouse with tumor, you wait until it's palpable, then you give a single dose of your different anti-CD25s, and you combine with anti-PD-1. This model is resistant to anti-PD-1. So this is the tumor growth. So untreated animals, they all grow tumors. If the animals are treated with a single dose of the old anti-CD25, the one that deplete in the periphery but does not deplete in tumor, you have no tumor control. Mouse is resistant to anti-PD-1. And when you combine this the old anti-CD25 with anti-PD-1, you have no synergy whatsoever because there is no depletion of the Tregs in the tumor. A single dose of the new anti-CD25, which can reduce fourfold the Tregs in the tumor, already gives you 2 complete responses and some partial tumor control. So we were excited about this. Something is going on within those tumors. And most importantly, when you combine with anti-PD-1, now you go from a completely resistant scenario to a very nice control with complete rejections of established tumors. So we got very nice synergy by improving depletion of this target. So this data demonstrate that CD25 is a good target and that antibodies with this and ADCC can mediate Treg depletion and promote therapeutic protection in combination with anti-PD-1. So at this point, we started collaborating with a biotech to try to take this into the clinic. And the first question was, is this the best we can do, right? So after 10 years of research, they asked us whether we could do a little bit better. So quite frustrating, but actually a very important question to ask yourself. So in thinking about what we wanted to develop, we wanted to develop a similar antibody to CD25 for the clinic. And the first question was, well, actually there are 2 antibodies, teclistamab and basiliximab, that have already been tested against solid tumors and they don't work very well. So how are you going to do better than that? Quite interesting. Those 2 antibodies, they're not very good ADCC. They're not very good drivers of depletion because they were developed instead to block IL-2 signaling onto T cells. And that's because they were designed to prevent graft versus host disease and also used to kill T cell lymphomas by depleting them of IL-2, this growth factor. And luckily enough, so complete serendipity, the PC61 antibody also blocks partially IL-2 signaling. And as I said before, IL-2 is a critical growth factor for activated effector cells. So we posed the following hypothesis. PC61 binds to CD25 on regulatory T cells, highly expressed there, very similar to CTLA4. The effector cells express CD25 but at very low levels. So our antibody, now that we fix the Fc receptor binding, can induce depletion of Tregs, but will not deplete the effectors because they express very low levels. Now, as I said, PC61 and most likely teclistamab and basiliximab will be blocking potential signaling via IL-2. And the first thing that happens when you kill Tregs, you have more free interleukin-2. And that free interleukin-2 could be a great growth factor for effector cells, but you are wasting it in the context of PC61 because you're blocking that interaction. So we said, well, let's design an antibody that binds to a different epitope so we can kill the Tregs and get that extra IL-2 to soup up my effector cells. It's an hypothesis, but it's testable. So we designed the new antibody and we tested it. So this is an IL-2 signaling assay. So we're just testing pSTAT5 downstream of T cells in the presence of PC61 or in the presence of the new antibody that we developed, which we call CD25 NIV, non-IL-2 blocking. So this is the in vitro data. So this is the maximum signaling via the cells in the assay, so 50% pSTAT positive. If you give the PC61, it blocks 50% of the signaling. If you keep block IL-2 with a bona fide IL-2 blocking antibody as a positive control, you completely lose signaling. And if you use the anti-CD25 and IB, signaling is intact. So we have an antibody that binds to CD25, mouse, and doesn't block IL-2 signaling. We did lots of in vivo signaling tests to validate this. The data is the same. So the piece of resistance is obviously to look at its activity in vivo against established tumor. These are mice treated with PBS after the tumors are palpable. These are mice treated with a single dose of the PC61 that depletes. So this is a depleter, but IL-2 blocker. And you see here 10% complete responses as a monotherapy, 10% delay in responses, and most of the tumors end up growing. So this antibody is the one that requires the anti-PD-1 to give you complete responses. Meanwhile, the new one, the anti-CD25 and IB, a single dose in tumors that are immunogenic gives you near 100% complete responses every single time we run it. So very powerful antibody. Question is IL-2 really doing the trick? That was our hypothesis, right? So we did blocking studies to really test this. So this is now the average of 10 mice, the growth in 10 different mice, showing a single curve. These are mice control. If you give them a single dose of the anti-CD25 non-IL-2 blocking, you have the complete responses that I described before. If IL-2 is critical, you should be able to ablate this response with an anti-IL-2 neutralizing antibody, and that would be the blue line. So you ablate it. So IL-2 is necessary. Now, it's not surprising that IL-2 is necessary, but what we want to demonstrate is that the blocking activity of PC61 is detrimental to single agent activity. So for that, we combine the anti-CD25 non-blocking antibody with PC61, right? So in this case, the PC61 will be giving you that blockade of IL-2, and you completely lose the anti-tumor activity. So with this experiment, we showed that, yes, it's the blocking activity of IL-2 on PC61 and likely in indaclizumab on basiliximab that prevents monotherapy activity of anti-CD25 agents or Treg depleting agents. So IL-2 is really doing the trick. And then we went into a series of experiments and combinations that you can imagine combine against everything. I think one of the, and yes, you get more and more responses. Now, the challenge always is against poorly infiltrated tumors. And in our case, the poor man's model for poorly infiltrated tumors is P16 melanoma. This is a model that in these conditions is resistant to GVAX and anti-CTLA-4. So for me, it was a pretty decent benchmark. GVAX is irradiated B16 that you inject therapeutically on the opposite flank to the primary tumor. And basically, what you see here is that the GVAX doesn't provide any protection. The anti-CD25 monotherapy as a therapeutic intervention is not impressive, but it gives you some tumor delay and some increase in survival. And when you give the combo, you actually really induce, except 2 mice, you induce complete rejection of tumors, but then later relapse. So you get resistant eventually with the combination of GVAX and anti-CD25. And what I like about this model is that you can do a lot of functional analysis or high dimensional analysis of the tumor microenvironment. I'm not gonna go in details on this, but it's just to show that you can go very deep into analysis and understand the independent contributions of the CD25 and the vaccine. And my mouse stopped working. And long story short is that what the CD25 really is doing is taking out a subset of Tregs, obviously, then a subset of CD4 cells that are PD-1 high that have been described as suppressives. The GVAX is doing something similar at the level of those suppressive CD4 helpers/suppressors, but most importantly is driving the infiltration by activated CD8 cells. And what the combination does is the same thing, plus the appearance of these populations 18, 19, and 4, which are NK cells. So it's really a synergy process. The mechanism underpinning resistance are something that we're looking into. So in the last 2 minutes, just to stress that our aim in the lab is really to translate our basic science findings throughout the collaboration with this little UK company, TAS Therapeutics, then that was later acquired by Roche, and we did a series of experiments and an antibody campaign to develop an equivalent anti-human antibody, and that antibody now is called RGS6292. Again, this was co-developed between my lab and these two companies. And the idea was an antibody that can target CD25 without blocking IL-2 signaling on human T cells. And we decided to go for a format that has enhanced ADCC, so an afucosylated antibody. This is the crystal structure of our antibody. So RGS binds to an area of IL-2 receptor alpha, CD25, that doesn't intervene with CD25 binding to IL-2. So IL-2 is in purple. The beta and gamma subunits of CD25, of the IL-2 receptor are in gray. And in yellow, you see CD25, and our antibody completely opposite phase. So it binds to the side we wanted. When you do signaling experiments in human PBMCs, you see here the maximum signaling via STAT5, it's around 90% in the assay. When you throw daclizumab as a control, the Tregs stop signaling, right? So very nice control. The effector cells also, they completely lose signaling, but when you throw the RG6292, there is no negative impact on signaling. So we have a good binder, non-blocker. Can this binder non-blocker deplete Tregs preferentially? So we took PBMCs from patients, we activate them in vitro, gently activate them, and we track activated CD4s and CD8s over concentrations of the anti-human CD25, and you see that the Tregs are the ones that are lysated. Meanwhile, the activated T cells only have very high concentrations that are affected. So we think we have a good depleter. Key one is to really validate this in human tumor samples. These are lung cancer samples from patients with cancer and colorectal samples. And you see here, this is the number of Tregs at the beginning of the, with a no antibody control. If you add our new anti-CD25, you have depletion of the Tregs. And if you do it with an FC silent, so you ablate binding to FC receptors, you lose depletion. So a pretty good, pretty specific Treg depleting antibody, doesn't seem to touch effector cells. So the last we performed, this is just trying to summarize a whole antibody campaign on a few slides, but we ran also humanized mouse models of cancer where we reconstituted mice with human CD34 progenitors, thymus, etc. And then we challenge them with a human prostate cell line, and we track Tregs and activated CD8 T cells in the tumor. And what you see here is several different treatments, kind of to benchmark, attempting to benchmark the activity of our anti-CD25. You see here the Tregs, variable, right, but high numbers of Tregs in the tumor. When you give the anti-CTLA-4 with an IgG1, you see a depletion of Tregs. When you give an anti-OX40 with a human IgG1, so depleter, you see depletion, and the anti-CD25 that we created, something very similar. What's critical here is the activated CD8 population. You see, the anti-CTLA-4 doesn't give you an increase on the activated CD8. So we think that is having a negative impact. The OX40, it's having something that is either similar or potential increase. But the anti-CD25, again, as a monotherapy in these models, with all the caveats that the model has, induces very nice Treg depletion and very nice activation of the effector T cell compartment. So all of this data plus a large tox package, I can't pass these slides, have supported the clinical trial initiation, which started, I believe, was end of November last year. And now there are more than 2 handfuls of patients that have been treated with this antibody, and we continue to monitor pharmacokinetics, Treg depletion, and we hope to start seeing some responses soon, so fingers crossed. Conclusions is I hope I convinced you that CD25 is a selective target for Tregs in mice, in humans, and even in the context of immunotherapy. You can optimize the mouse antibody to now have a proper mouse surrogate to use in combination experiments as a therapeutic depletion. And then we were able to develop this next-gen anti-CD25 non-blocking antibody, which has supported the development of this human antibody that is now in phase 1. So this is my team, really a phenomenal group of collaborators. Marta, who started the engineering of the antibody, Fred, who led the first generation anti-CD25, and Isabel, who led the work on the second generation non-IL-2 blockers. The TOS team led by Anne Gubier, and then the Roche team, who overtook task, who is led by Maria Mann, Kerry and Martin, who we learned from whom we learned antibody engineering. We knew nothing about it, and the team of Daryl Marsden, always very supportive with the clinical translation. Thank you very much for your attention.
Thanks, Sergio. That was great. A real tour de force. Sergio, can I start by asking you a question about the clinical trial, which seems permissive for all patients with advanced cancers? Do you have any data from the samples that you've looked at from the clinic as to which tumours you might be most hopeful to see a response in?
Yeah, so this was probably a year's discussion with them. If you look in mouse models, right, and this goes to the question that I asked before, my mouse models tend to be Treg heavy, the immunogenic mouse models. And they tend to be Treg heavy because they infiltrate following the wave of effector cells infiltration. And the CD25 works as a monotherapy on those highly infiltrated tumors. So the obvious suggestion for us was to go after those. And we have done quite a bit of analysis at UCL on clinical samples. So the ones that we suggested were ovarian. I think that's a very interesting one, very packed with Tregs.
Yeah.
Lung cancer, melanoma, and head and neck. Those were on my suggested list. I can't disclose exactly what they went after, what they have been favoring, but it's a basket, right? But those are the ones that we have plenty of data to say you have lots of effector cells and lots of Tregs kind of counteracting those. I would suggest that the idea would be to go after those tumors in combo with a PD-1. But obviously, what you want in the learnings of the IDO stories and all that is to really have a clean arm of monotherapy.
Yes. So questions come in, Sergio, for you about is the drug being trialed as monotherapy? Isn't there a comparison to IPI?
No, right now the trial is not being run in comparison to IPI. We have funding for a trial with Paul Mulholland in GBM. It's a neoadjuvant trial. I think it's going to be a wonderful trial where we have an arm of EP, and we've been in discussions with Roche for them to give us anti-CD25 for an arm of anti-CD25. So if that happens, which will be tricky, right? Because they're still at the dose finding exercise. I think this is going to happen, but later on. But when that happens, that's probably going to be the first head-to-head benchmark of EP and CD25.
Okay, thank you. We're running a little bit behind, but I'm hopeful that we'll be able to catch up. Alan, can I ask you to share your screens? Thank you very much, Sergio. That was great.
Thank you. I'm trying to stop sharing. Oh, there we go.
Can you hear me okay? And is that now sharing?
Thank you, Alan. Please take over.
I hope I am. So thank you very much, and thanks for the invitation to talk today. And actually, I'm going to pick up very much on what Tim was saying about, in context of radiotherapy, understanding as well as we can what the immune system does when you give radiotherapy to patients, because actually very, very little is known about that. And we took the opportunity of rectal cancer as an example of how we might learn more. This is not about trials of combinations at this stage. It is simply about understanding what the biology of the immune response is when we give radiotherapy to patients. And I think this is a first start on the sort of themes Tim was talking about in terms of their plans in Manchester, which certainly to some degree we share, about looking across different cancers. And this starting point was in rectal cancer in a project done very much in collaboration with my old hunting ground in Leeds. So this is very much others' work, as so often, and so Elisa Fontana and Anna Wilkins and Angaraj Sadanandam really did all the work that I'm going to talk about. And I think both Anna and Angaraj are listening to this, so I better do all right, hadn't I? So thanks to them. And all of this is about rectal cancer, and I used to be a GI clinical oncologist in a previous life. And the advantage of rectal cancer is we really are taking an opportunity within standard of care radiotherapy. So patients with rectal cancer are quite often given a course of radiotherapy before they have surgery. And what that means is that you can compare and potentially collect samples where pre and post treatment. So when the patient is first diagnosed, they need a biopsy. That gives you a sample. Then they have their course of radiotherapy, and I'll come back to what that comprises a little later. And then they have a big operation to take out that tumor after that operation. So we have a rather unusual, ready-made, unique opportunity, not unique, but an opportunity to look at pre and post radiotherapy in the clinical context and look at what the immunological consequences of that intervention is. And we know that patients have a variable response to that preoperative radiotherapy. So a key question is, how do you measure that response to the radiotherapy? Because you can do that by looking at the biopsy and then looking at the surgically resected specimen. And that has been done for many years in various ways of pathological response, particularly tumor regression grade. But in Leeds, Phil Quirk, Nick West, and team David Sebag Montefiore on the pathology side developed this tumor cell density score, which essentially is a more quantitative analysis of the response than a pathologist looking and saying, well, this looks good, bad, or intermediate. And this works by putting a number of arrows, as you see on a graticule, and scoring whether the end of that arrow hits a tumor cell or not. And that then generates a score of a tumor cell density expressed as a percentage. What we were able to do using this score with Nick's help was to essentially divide the responders into a poor, intermediate, and a good group, with the ranges you should see of the change in tumor cell density. So a very particular clinical readout score. And what is very useful and important in rectal cancer is that we know that the better you respond pathologically after neoadjuvant radiotherapy in this context, the better your outcome will be in the longer term. So this gives us a very nice, early, robust measure of whether you are doing real clinical good to the patient rather than just improving their pathology. So this was the group that had been collected in Leeds. I won't go through it in detail, but what it essentially allowed us to do was a couple of analyses. I should note and absolutely concede, and we recognize that the numbers here are small. We won't make too much of them, but nevertheless, they'll show some interesting data as I'll come to. But in brief, we compared both we looked at both short course radiotherapy and long course. And what I mean by that is, depending on the clinical context, patients either get short course radiotherapy, which is 25 gray in 5 fractions over 1 week, followed by early surgery at 1 to 2 weeks after radiotherapy completion. And in contrast, there is also a long course radiotherapy course, which is 45 gray in 25 fractions over 5 weeks, given with concurrent chemotherapy. And in that case, the surgery is delayed to between 4 and 6 weeks. I haven't got time to go through the clinical reasoning, and actually some of those parameters are now changing. But nevertheless, this was the cohort we were looking at. And in brief, accepting that the numbers are small, we did not see major differences in the short course and long course outcomes that we looked at, but recognize we need to look at more samples, which we plan to do to differentiate that difference. That comes back to the question of whether dose fractionation matters, but at least in this current analysis, we can't pick those factors apart. But what this group did allow Anna and Anne Garage in particular to do was a couple of things. So firstly, they could look at the baseline sample and see how that correlated with the pathological response at surgery. The second thing they could do was look at the change in the immune microenvironment in the tumor before compared to after that radiotherapy, and again, see, did that link to the pathological response, yes or no? The other thing I should say is that this analysis is restricted to gene expression analysis and restricted to nanostring, and I'll come back to the genes and how those come about. But by nature of the fact that this was relatively old fixed tissue, the level of analysis we could do is not as strong as it could be with better samples. And again, I'll come back to that. So here's just a couple of examples of a poor and a good radiotherapy response, comparing before and after radiotherapy. And here you can see that there are lots of tumor cells still after radiotherapy, and that change in TCD was actually slightly positive. Here's a good responder, tumor cells very much gone, replaced by stroma, and a lot fewer tumor cells after that radiotherapy. And one of the first things that Anna could do, which I think is interesting, again, we need to learn and address more questions, was that by the TCD technique that Nick developed, you can quantify the stromal relative to the tumor density in a sample. And what we saw was that the more stroma there was in the tumor before radiotherapy, that correlated with the outcome in good versus poor responders. Moving on, however, to what most of this study was about, which was around gene expression, was in these fixed tissues, although even particularly the biopsy specimen for diagnosis is relatively small, what you can do is macro dissect out the tumor that you can see. You can extract RNA and you can perform a focused gene expression profile. And from NanoString, there are 770 immune genes, which, as you can see, cover the immune response, which we looked at. We also spiked in another 30 genes to try and look at the DNA damage response in these same samples, although I won't talk much about those, and they didn't really show anything very significant. So the real focus here is on the immune gene expression profiling. And the first thing that you can do is to look at the pre-radiotherapy biopsies and see whether the initial gene expression did or did not link to the good or poor response. And you can see quite clearly that it did. Just the pattern of these immune genes and their expression was different in the good versus the poor responders. And in particular, those genes which were upregulated in the poor responders, again, at baseline, are the list that you see here. But importantly and interestingly, they are some targetable genes. So particularly PD-L1, the higher the PD-L1 expression in your pre-treatment sample, the worse you did in the pathological response outcome. And that provides, if you like, a rationale for combining anti-PD-L1 or anti-PD-1 blockade with neoadjuvant chemoradiotherapy, which is something that is being done. But to be honest, those combination strategies, to echo what Tim was saying earlier, have tended to be rather scattergun rather than biologically informed, but at least this is a signal that PD-L1 blockade might be a useful way to go. The other thing you can do from these gene expression profiles is to deconvolute them into what cell types they are likely to represent in the pre-treatment biopsies. And again, how do those correlate with pathological outcome? So these are the sorts of populations which were upregulated in the poor responders. And again, interesting to see that some of the cell types, both at the top and in a little more detail at the bottom, that Tim was outlining and targeting in his preclinical models, these are also coming out in some of these analyses, again, providing what I think is a critical link between mouse modeling and human data to, if you like, boost the strategies that we should be optimizing and going after. But again, the myeloid population, the monocyte population, macrophages are a sensible target from our data too. So what about now the second part of the analysis that I mentioned, which is all about whether the gene expression changes before and after radiotherapy and whether that links with pathological response. So there are some expression profiles out there. For example, this is the T-cell inflamed gene expression profile, which has been looked at, developed and validated by others in different human clinical contexts. But what you can see here is on this particular profile, if you upregulated this profile, you tended to respond well and you could see a difference, whereas in the poor responders, no difference was seen. So this goes back and links, if you like, to some of the things about if you turn a cold tumor hot with your intervention, you will do better, and that seemed to be consistent with our findings. And here in a bit more detail is the heat map of those and the genes and the types of pathways that they represent. And again, you can see a significant change between the biopsy pre-radiotherapy and the resection post-radiotherapy in the good responders, but you do not see that in the poor responders. So the immune development and change with radiotherapy does seem to be clinically important. Then in the final part of this, what Angaraj in particular has done is to take this one step further. So rather than looking at those individual genes in the heat map and seeing whether they go from blue to red, what you can do is what Angaraj can do is to link these gene expressions up into pathways. And on the left, what you can see is the pathways that again were upregulated during treatment and by radiotherapy, which signaled signs of a good response. And again, perhaps reassuringly, right at the top of that is the interferon gamma response, which is also shown on the right there. So that's reassuring, but so many other interesting pathways which come out of other types of biology, but give us a signal of where we might want to go. But one of the ones I just want to pull out that is most interesting, perhaps, is that actually when Angaraj looked, one of the 5 most significantly upregulated pathways, when put together as a network, these very much mimic the type of immune response you would expect to see in relation to a viral infection. And so what that is suggesting is that when you give radiotherapy, if you trigger a virus-like immune response, that will be a signal of a good clinical outcome. And this at least is consistent with what others have shown in some other contexts. This is an example of Sandra Damaria's trial, in this case in non-small cell lung cancer. They were giving radiotherapy with anti-CTLA-4, and what they found was a rise in type 1 interferon in the blood, again, was linked with a good clinical response. So these are all bits of evidence and areas that we now want to pursue in more detail, that there's no question that the immune response to radiotherapy is complex, and many different types of immune response may be triggered, and it's not going to be a single story. And the stromal element and all of those things will play into that. But are there signals that we can look for which may be a good response? And the virus signal is one that we are particularly interested in. And what I would say is that in recognition of all the uncertainties of questions about doses and radiotherapy fractionations, whatever we find out about the immune response to treatment, we're not going to suddenly drastically change our clinical practice on the basis of that. So what we have to do, and the rectal cancer data I've just shown you is an example, I hope, of that, and Tim echoed it too, was to understand more about what we're doing within standard of care radiotherapy in the immune context, and build on that to develop rationally informed clinical trials. So just in terms of next steps, we clearly need more numbers, but I think we've generated interesting signals even from this relatively small data set. The dose fractionation question we've mentioned, and again, rectal cancer is an interesting example because standard clinical care includes 2 very different doses and fractionations. We clearly need prospective studies as well as retrospective, and we have plans along the lines that Tim is also doing in Manchester to do that across different tumor sites. But if we do that and we can get more tissue, potentially fresh frozen, we can look at the blood. There are many other things that we can look at from an immune perspective. In this study, we've looked at gene expression, but we could also think of mutational burden before and after radiotherapy, TCR repertoires, and imaging. And then we can take the sort of findings that we get to address questions back into the preclinical murine models. And just as an example of the imaging things that we're starting to do with the Vectra machine, this is an example of a short course radiotherapy patient where here's the pre-treatment biopsy. We can look at the tumor cells with cytokeratin, but some of the other immune markers that you see. And here, this is a good responder. And in the post, even after short course radiotherapy with early surgery, you see that the tumor cells are looking pretty sick after this treatment. But again, here we can see lots about the immune cells, and what Vectra and other technology allows us to do is to now, what we're doing is to build up the quantitative analysis of this, not only what types of immune cells there are, but where are they in relation to the tumor? Those are the sorts of questions we can now build on to the first data set as I've discussed today. So in summary, poor response, we believe you may have a lot of stroma or a lot of fibroblasts, more myeloid populations, and we need to understand much better the biology of what is happening between these 2 groups. And the tumor cell density has been a very powerful way of looking quantitatively about an early pathological response endpoint, which is very clinically powerful in this particular clinical context. And you need things to become hot during radiotherapy. That's what our data suggests to date, perhaps more specifically with a viral type signal if you want to do better in terms of your clinical outcome. So finally, just to say particular thanks to many people, but Anna, Elise, Angaraj, and Nick for their help with this data. And I'll stop there, and thank you very much.
Thank you, Alan. That was great. Very significant progress in giving us some leads. Can you say a little bit more for us about the correlation between the stromal density correlating to the radiotherapy response? do you have any clues about what it might be within the stroma that's important?
A good question. Honest answer is no, not at this point. That was to be honest, an incidental thing that Anna spotted. She just noticed that there was, you know, from the tumor cell density, which is purely a, you know, putting arrows on the screen type approach, there was more stroma in those who responded worse. But as you say, it opens up a lot of other questions which are addressable. Staining those cells with Vectra or with, you know, even more detailed analysis with a, you know, it's imaging site-off or codex. Also doing things like nanostring DSP, digital spatial profiling. We can actually go in perhaps and look at that stroma in the sort of way we have on a gene expression level. So a lot further questions, but at this stage we do not know.
Another question has come in, Alan, about did you see whether the interferon gamma signature was originating from the immune or the tumor cells?
Yes, good question. So the honest answer is no, we didn't. We would expect it to be the immune cells, not from the tumor cells. But again, that's the sort of thing that more detailed analysis should allow us to pick apart in more detail.
Thank you very much. So we'll move on. It's a real pleasure to introduce Professor Amato Giaccia, who will talk to us about the work that they're doing on immunologically resistant tumor clones. Over to you, Amato.
Great, thank you very much for the invitation. It's great to be here. Great talk so far today. So most of the work I'm going to tell you about was done or started at Stanford, and this is some of the work we're also working on at Oxford, which I came to last year. So many preclinical models that are used to study the immune response and radiation immune response rely on genetic modification. So by nature, they're not very immunogenic, but through the addition of either viral antigens or albumin antigens, they become now immune responsive. And then, so the question you have to ask is that, is this altered immune responsiveness really going to be informative then of what goes on in the human or spontaneous model system? So for these reasons, we decided to look at spontaneous tumors, and we started out with the mouse polyoma middle T-driven tumors in C57 black 6 mice, which developed breast cancers. And the cartoon shows that there was a tumor. We took tumor, we dissociated them, and then we derived tumor clones. And I will focus on 2 clones today, one called polyoma 117, the other on 119, one being highly responsive to radiation immune therapy, the other not. So here is a tumor growth delay of the 117 on the left-hand side and the 119 on the right-hand side. And what you can immediately see is that giving these tumors one single large response, the 117 are highly responsive. They exhibit a precipitous drop in tumor volume, and then because the dose of radiation is not sufficient to be able to sterilize all the tumor clonogens, they start to regrow. Whereas if you look at the 119, and giving the dose at the same time, is that they exhibit a rather brief delay. There's no precipitous drop in volume, and they continue to grow. And this is really reflective of a situation that occurs in the clinic. So what is mechanistically occurring here? Well, it's not due to changes in intrinsic radiosensitivity. If you do a clonogenic survival, in fact, the sensitive 117 is more radiation resistant in vitro than the resistant 119 in vivo is in the survival curve. So not a difference in intrinsic radiosensitivity. In addition, there's not a difference in microenvironmental factors. But if you implant these tumors in immune deficient mice, what you see is that the 119 are resistant regardless, there's no effect. But the 117 tumors, they lose their sensitivity in this immune deficient background, in the nude mouse background, suggesting that the reason the 117s are sensitive is due to this combination of radiation and immune response. So if we actually look now at immune infiltration, and particularly CD3 positive T cells, in the 117, you can see that even without radiation, there is this immune inflammatory phenotype, this hot phenotype that Alan just so very eloquently told you about. Whereas in the 119, these tumors are rather devoid of T cells. And after radiation, there's even more in the 117, and there's still no increase in the 119. So if we now look at this in a little bit more depth, you can see here is CD45 cells that increase in the 119, as well as, increase in the sensitive tumors and go down in the 119. And you can see here is that if you look at CD45 positive cells, you see that there's really no real change in the 119, but an increase in the 117. If you look at CD8 positive T cells, they increase in the sensitive, CD4s, mild increase, and immature myeloid cells actually decrease in the 117, and macrophages increase in the 117, and to a lesser extent in the resistant 119. But the big changes is that in the sensitive 117 cells that are radiation sensitive, CD8 positive T cells go up. In addition, if you look at MHC1 levels, which are very essential in terms of being able to develop this immune response, is that they're very high in the 117 cells and they're extremely low. This is actually almost a tenfold difference in the level of MHC1 expression. And you can see this, if you look at PD-L1, you can see that PD-L1 levels increase with radiation dose in 117, but have no change in the 119. So if this is true, then how does the combination of radiation and PD-1 immune therapy alter the response of these tumors? So if we look at the top panel, we look at an isotype treated control, PD-1 treated, 12 gray treated with isotype, 12 gray with PD-1 and 12 gray with PD-1 plus CTLA-4. And what you can see here is that there's, except for radiation, there's no real response of PD-1 alone. Radiation, there's this precipitous drop again, but a tumor growth delay. If you add PD-1, you now see that about 25% of these tumors are cured. And if you add both PD-1 and CTLA-4, about 75% of them. In contrast to the bottom panel, no matter what you do, whether you give radiation alone or radiation plus immune checkpoint inhibitors, these tumors still continue to grow. And if you want to look now at the sum totals, here's a comparison of what's going on in the 117. So this is survival now, looking at it a little differently. So the 117, what you can see here is that if you give PD-1 or CTLA-4, you've got now long-term survival of almost 75% of these animals compared to no treatment alone or radiation alone. And if you look at the 119, again, no matter what you do, is that almost all of these animals are dead by 40 days. So 117 tumors responsive to radiation and immune checkpoint therapy, 119 tumors resistant and refractory to both. So this is the real question then. 8119 tumors represent a model of therapeutic resistance. They exhibit low levels of MHC1 and they do not exhibit a T cell recruitment and activity in their tumors. So how can we overcome this? So if we analyze these tumors, and this is reverse phase protein array, is that one thing that became very obvious was that 117 tumors were highly epithelial in origin compared to 119 tumors. Here is a RPA looking at cytokeratin and a number of EMT markers, and you can see very dramatic differences. And that if you look at the gene expression level changes, that one that was very noticeably different, to our surprise, was called AXL. AXL is a receptor tyrosine kinase, with the 119 tumors having very high levels, which don't change with radiation, at 117 levels having lower levels. And then if you want to look at this on a FACS analysis with a log-based scale, you can see that 119 tumors exhibit very high levels of AXL expression. So AXL belongs to this TAM family of receptor tyrosine kinases. And what's interesting is that AXL, one of its major, if not most prominent roles, is in epithelial mesenchymal transition. Regulates invasion and metastasis, drug resistance, desmoplasia, and it plays a role in senescence. So if we now look at the effects now of AXL in a more rigorous genetic manner, and that is using CRISPR to knock out AXL in these resistant tumors. And you can see here is that we're very efficient in terms of AXL reduction using CRISPR knockout. And if we compare what that effect is, here is the mesenchymal phenotype of the 119, the epithelial phenotype of 117. If you just use a empty CRISPR vector alone, no effect. If you now knock out AXL using CRISPR, you can see you've transformed the mesenchymal phenotype into the epithelial phenotype. Okay. So what effect does that have in terms of intrinsic radiosensitivity? And the short answer is none. So if this is in vitro survival, just altering AXL expression did not alter in vitro radiosensitivity. However, just altering AXL expression alone had a profound effect on tumor growth. So the red are the controls, the blue are the AXL knockouts, and you can see that just inhibiting AXL alone was able to induce a diminishment in tumor growth. However, if you combined AXL knockout with radiotherapy, you saw you got a very profound tumor growth delay in these radio-resistant 119 tumors. And that this effect, in fact, was immune responsible. So that if you do the experiment by injecting these animals into immune deficient cells, you can see now that just losing AXL, which restored the radiation response, resulted into now a lack of radiation response when implanted into the nude mice. So what was going on at the immune level, you can see now that in these CRISPR knockouts, you saw an increase in CD45 T cells, saw a decrease in macrophages. You saw a decrease in these immature myeloid cells. And most importantly, you saw an increase in CD8 T cell infiltrates. So at the molecular level, we saw that MHC1 levels were restored when we knocked out AXL, and that we think a great deal of changes actually occur at the level of the NF-kappa B transcription factor, which was very telling because you can see that downstream effectors such as IL-6 decrease now in these cells. So MHC levels went up and NF-kappa B levels went down. And this decrease of NF-kappa B correlated with a decrease in a variety of growth factors and chemokines that we thought were responsive for this both radiation and immune suppressive phenotype. So you can see now that CSF1, CSF2, CSF3 all went down. the CCLs went down, especially CCL5, and that the TNF, IL-6, and IL-1 alpha also went down as well. So immune suppressive modulatory factors, when AXL was knocked out, went down in these 119 tumor cells. So I said to you that one of the major roles of AXL has to do with EMT. And what this shows is that we're looking at AXL in a polyoma signature, but more importantly, AXL at an EMT signature. And you can see that AXL corresponded very nicely with this EMT signature. And this is an EMT signature, not from mouse tumors, but an EMT signature in human breast cancers. So in these human breast tumors, which have very high EMT signatures, there's a very strong correlation with AXL expression, suggesting that in this subset of human breast tumors, treatment with anti-AXL therapy would not only alter their EMT signature, but would make these tumors much more radiation responsive. In addition, you can see that not only were these 119 tumors more responsive to radiation when AXL was knocked out, they're more responsive to immune checkpoint therapy as well. So this is a series of experiments that look at what happens if you inhibit expression of AXL in tumors, but what if you inhibit the expression of AXL systemically? So here's MCA 30 tumors. They're AXL deficient, and we implanted them in AXL deficient mice, or CD57 black 6 mice. And what you can see is that if you implant the AXL tumors in the C57 black C, the wild type, you can see that they grow very nicely. These red lines represent individual tumors. But in the AXL deficient mice, there's a very significant inhibition of their growth. And that this also occurred in a number of other tumors when implanted in these AXL deficient mice. So what's going on? Well, the AXL deficient mice have lower levels, have higher levels of dendritic cells, and they have much higher levels of CD8 positive T cells. So these are constitutive knockouts. The whole mouse exhibits AXL deficiency throughout all of their cells. So is this increase of CD8 positive T cells going to be sufficient to explain what's going on? Well, if it is the case, then eliminating these CD8 T cells in this AXL knockout background should then make these tumors refractory in terms of growth inhibition. So here, what we're looking at is tumor growth in the AXL deficient cells in black. If we now immune deplete these AXL deficient cells of CD8 positive T cells, they grow just like their C57 black 6 wild type parents. And in fact, if you do the same experiment in the C57 black, in the black 6, you see that there's very little effect in terms of tumor growth. So this experiment tells us that we may not necessarily need to inhibit AXL in the tumors themselves, but that systemic treatment of AXL should be sufficient to be able to infuse tumors systemically with CD8 T cells. So this is a little cartoon summarizing what I just told you about, that here we have these mesenchymal breast tumors, and that they have high levels of AXL expression. And those high levels of the AXL expression inhibit MHC and cause the increased expression of cytokines and immunosuppressive growth factors that result in elevated levels of macrophages. That if we now inhibit AXL expression, we get an increase in MHC levels and a decrease in these macrophage levels. And most importantly, we get an increase in CD8 positive T cells and responsiveness now to both radiation therapy as well as T cell checkpoint therapy. So what I showed you mostly were genetic studies that we could control in a very rigorous manner. But I would like to tell you that, that this is hopefully going to be tested in the clinic very shortly. In fact, we look forward to potentially joining with our colleagues here throughout the UK in testing this. We developed a soluble AXL decoy receptor, which has got a very potent genetically enhanced affinity to inhibit its only no ligand. AXL itself is very rarely mutated in cancer, and therefore we think that this is going to be probably the most direct and sensitive manner of inhibiting AXL signaling. And so we'd like to be able to take this into clinical trial and look at this in terms of combining it with radiotherapy and T-cell checkpoint therapy. So this decoy receptor is going to go into phase 3 clinical trial very shortly in metastatic ovarian cancer. And it's got a rather excellent safety profile in that there is no adverse effects or grade 3 or grade 4 toxicities that have been observed. So very exciting times to be able to translate some of the analysis from preclinical models into the clinic. So I will just end with this slide. In particular, I'll just call your attention here to Todd Aguilera, very talented fellow in the lab who is a radiation oncologist and who's now moved to Southwest Medical School and set up his own laboratory. So I'm very happy to answer questions, and I thank you again for your attention.
Thank you very much, Amato. It's great. Can I ask you about the dose of radiation that you use with the 20 gray? Do you see an effect with lower doses of radiation and/or fractionation? My first question. And the second is, have you reproduced these results in other tumor types, and have you reason to think they might be different?
Yeah, so we've done, those are a lot of questions, Tim. Yeah. We've done this at lower doses. We've done 12 and 15 gray. And we've also done this at 3 times 3 gray. In terms of other tumor types.
Does it make a difference, the different doses?
Hard to tell. I mean, does it make a difference? You don't change the response of the resistant tumors and the radiation responsive tumors are still responsive.
Okay.
To both radiation and T-cell checkpoint therapy.
Yes.
In terms of different tumor types, we've looked at a number of different tumor types. So we've also looked at ovarian, and we've just started rectal tumors as well.
Are there any other questions for Amato?
Can I ask a quick question, Tim? It's Crispin Hiley here from UCL. Thanks so much. That was fascinating stuff. Do you know the sort of mutational burden of these tumors? Is it a sort of highly mutated tumor? And have you sort of done any work to look at what the antigen is that the CD8 cells are responding to?
Yes, very, very good question. So we looked at the mutational burden. It's not very high. In addition, both the 117 and the 119 tumors had the same level of mutational burden. So the mutational burden could explain their response to CD8 or T-cell checkpoint therapy. And in terms of the antigen, we don't know.
It gives me enormous pleasure to welcome everybody back, and in particular to welcome Professor Kevin Harrington, who will be talking to to us about combining DNA damage response modifiers and radiation to modulate the anti-tumor immune responses. Kevin, thank you very much.
Well, many thanks, Tim. And so here is the title slide with some of the recognition of the funders of this research. These are my disclosures in relation to this and other research. So the first thing to say, and of course I'm drawn to the name at the bottom right-hand corner here, is that the work I'm going to present this morning in the main deals with ATR inhibition and the potential interaction that has with the immune response. And the data that I'm going to show is generated by Magnus Dillon, who did his PhD in the lab and has recently been appointed to a clinician scientist position at the ICR and the Royal Marsden Hospital. And I will show some unpublished data at the back end of the talk from a postdoc, Emmanuel Patin, who works in the lab. So for those who don't know about ATR, ATR is one of the major components of the DNA damage response and cell cycle control. And it responds largely to RPA-coated single-stranded DNA, leading to downstream signaling through CHEK1, leading to cell cycle modification at the S phase, where it's involved in dealing with replication fork stability, and also at G2M checkpoint, where it's involved in the cell cycle checkpoint. And there are a number of publications, and just a few of them are highlighted on the left-hand side of the slide, indicating the importance of this pathway in various aspects of radiation and response to chemotherapy. Now, in Magnus's PhD, he initially demonstrated, and we were interested at this time, in ATR inhibition as a standard radiation sensitizer, if you like. So essentially, what Magnus shows here in a publication from about 4 years ago is that at modest doses of the ATR inhibitor, AZD6738, now known as ceralasertib, you can see that there is radiation sensitization with sensitizer enhancement ratios of between 1.5 and 2 to single fractions of 2 and 4 gray of radiation. In the bottom left-hand side, and I'm not sure if you can see my mouse, in the SCC7 spheroid assay that Magnus developed, you can see that when we give fractionated radiation of 20 gray in 10 fractions, with or without the ATR inhibitor, you see profound radiation sensitization to fractionated courses, which is mirrored in the mirror panel and in the middle panel in vivo studies. And you can see there the Kaplan-Meier to the right-hand side of the slide. There had been a hypothesis that this sensitization would be predominantly seen in P53 mutant tumor cell lines, and also potentially in cells that were defective in DNA damage response, such as through BRCA2. And what Magnus demonstrates here is actually that that is not the case. And you can see that for the paired cell lines that are P53 proficient versus deficient, or BRCA2 proficient versus deficient, there is still a sensitization event, with the P53 mutants being a little more resistant and the BRCA2 mutants being a bit more sensitive to radiation and to the combination. And in contrast to data from Esther Hammond and others, that maybe this would be more profoundly seen in areas of hypoxia, we did not see an increase in gamma H2AX focus formation, at least, in oxic versus hypoxic environments. We have shown that the radiosensitization depends on cell cycle progression, and more of that later. So again, along the top, you can see in the clonogenic survival assays for a range of tumor or transformed cells, Cal27, MCF10A, and the retinal pigment epithelial cells transformed in hTERT, you can see that there is sensitization with ratios of between 1.47 and 2.16, whereas in mouse fibroblasts, you see that there is less of a sensitization effect. When you look through those middle panels across the center of the slide, you can see that the effects on cell cycle distribution in those cells which are malignant and deficient in G1S checkpoint control, you can see that the predominant effect of radiation is cell cycle arrest in G2M, which is not seen in the normal mouse fibroblasts to the right-hand side of the slide. And as a consequence, when we give the drug, the ATR inhibitor, and we look at the comparison in cell cycle distribution after 10 gray of radiation without and with the drug at 16 and 24 hours, you can see that we can abrogate that G2M checkpoint arrest. And in the bottom of the slide, what I show you is progression into mitosis on the left-hand side, measuring the percentage of cells showing staining for phosphohistone H3, a marker of mitosis. And you can see that radiation reduces that progression into mitosis, and the drug AZD6738 in a dose ascending fashion mitigates that arrest, abrogates it, and causes cell cycle progression. And furthermore, if we look at radiation survival under serum starvation conditions where the cells are arrested, we lose the sensitization effect of the ATR inhibition. If we look in vivo at the numbers of unresolved gamma H2X foci in tumors extracted from treated animals with HCT116 tumors, you can see that that is increased under the drug plus radiation conditions. And indeed, when you look at the formation of Rad51 foci, the hallmark of homologous recombination repair, you see that that is reduced when the drug is on board with radiation. Now, to the right-hand side of those histogram data, I show some microscopy demonstrating the generation of micronuclei, and I'll say more about that in a few moments. Again, you can see in the Western blot, there is, as you might imagine, markers of cell death, and we can see markers of apoptosis, for instance. Now, these are time-lapse microscopy data using the Fuchs cells, so the fluorescent ubiquitinated cell cycle indicator cells. And you can see in the color coding along the top panels where the cell cycle distribution is demonstrated, that in response to radiation following the green curve, you see, as anticipated, a prominent G2 arrest in these treated cells, and that is not present when the AZD6738 is on board. And in contrast, when you look to the right to look for the S phase fraction, you can see that when the ATR inhibitor is on board, cells are progressing more often through cell cycle. And again, we were able to quantify the numbers of micronuclei that we could see on these serial confocal microscope images. And you can see that the drug plus radiation is leading to a profound formation of micronuclei. Furthermore, we were able to see a number of patterns of cell death, and I illustrate for you here in these color-coded cells an interphase death and a more classical mitotic death, green cells being cells in mitosis, and you can see the bubbling up of that cell in the mitotic death panel. At the bottom of the slide, you can see some of the micronuclei staining for gamma H2AX in the top panel, with the counter stain being DAPI, the dark blue. And along the bottom, you can see that the cell cycle distribution, very interestingly, of these micronuclei doesn't always comply with that of the main nucleus. And I have the name here, Martin McLaughlin, to remind me that Martin McLaughlin, who will be hopefully shortly starting his own laboratory in Belfast in the near future, was involved in all of this work and was instrumental in delivering this work. So we and others were very interested by back-to-back publications in Nature about 3 years ago, looking at the effect of micronucleus generation on inflammatory signatures and paracrine signaling. And of course, studying ATR inhibition, the understanding that cells that are prevented from performing an effective G2 block and who are forced into mitotic progression with unrepaired DNA damage may be more likely to generate these micronuclei and potentially act as signaling platforms for CGAS sting. So I segue briefly, and I'll come back to this in a little time, some data from a clinical trial where, again, led by Magnus, who was the clinical fellow delivering this trial. Here you see a patient with a very unpleasant recurrence of squamous carcinoma on the front of her neck, as indicated by the white arrow, with a partial remission to the ATR inhibitor. And from biopsy specimens before and on treatment with the ATR inhibitor, we saw significant changes in PD-L1 expression. Now, this led Magnus to want to study the immune effects of ATR inhibition in the context of radiation. And again, here you can see an immunocompetent animal model now, TC1, potentially a head and neck model. It's really a lung cancer, but it expresses E6, E7 from HPV. And you can see that there is radiosensitization in response to a modest dose of radiation, 8 Gray in 4 fractions. But what Magnus and Martin McLaughlin did was that they looked at the immune cell infiltrates within the tumors. And again, what you can see here, and these are deconvoluted data from nanostring analysis, you can see that there are profound changes in a range of different, this is a logarithmic scale, in a number of these cell compartments. And I draw your attention in terms of the good players to changes potentially in the T cell populations, but bearing in mind the Tregs are going up. But also look here at the NK cells, and I ask you to store that piece of information for the future. And note also that macrophages and potentially some MDSC-like cells are also potentially going up under these circumstances. Looking again at the nanostring data, we were able to look at pathway enrichment, and this is the color coding for this graph here in the top left of the slide. You can see that there are profound changes in gene expression within the irradiated cells that have ATR on board. And you can see a number of the pathways listed down the y-axis of this graph indicating what's going on. You can see here, just as a highlight, changes in PDL1 expression, and I draw your attention to the cytokeratin positive cells where there is a modest change versus the other components, which are the immune cell components, where there are more profound changes. This is the color coding here, so the blue indicating the combination of drug plus radiation. Again, we looked through the nanostring data and also through a protein array. We looked at changes in gene expression and protein expression of immune signaling agents. And you can see here for the ATR plus radiation that there are profound changes both in vitro and in vivo, again using the TC1 model, borne out again using the protein cytokine array. And when we look at cytokine signaling, in an experiment that was requested by reviewers, and in fact was a very helpful comment from reviewers, what we did was we were able to sort both the tumor cell population, the CD45 negative, and the non-tumor population, including the immune cell compartment, CD45 positive, indicating profound changes in a number of transcripts. And I draw your attention particularly to the fact that the tumor population is signaling with a number of agents that may well be stimulatory to and attractant to components of the immune system. We and others have been interested in the potential role of activation of pattern recognition receptors, including DNA and RNA sensing moieties within the cytoplasm of cells. And here on RNA data, I show you for sorted cells again, so this is from the tumor cell compartment, that you can see that there is upregulation of RIG-I, MDA5, and other signaling components, including ZPP1. There's no change in STING transcripts. That doesn't necessarily mean that STING isn't signaling better. Now, when we turn our attention to what's happening in the antigen presentation, processing and presentation machinery, you can see in the differentially sorted cells, so again, the CD45 negative versus CD45 positive populations, again at transcript level here, you can see that there are changes in transporters into the endoplasmic reticulum, in components of the immunoproteasome, and in class 1 expression in tumor cells, and in class 2 expression in the non-tumor cell compartment. And this is borne out when we look, for instance, at FACS analysis on the right-hand side of the slide, looking at median fluorescence intensity on the cytokeratin positive cells. You can see that there is upregulation of MHC class 1 molecules, again, in keeping with something that would be consistent with an adaptive immune response. I change direction now, and I'm going to show you some unpublished data, and I'm going to recognize the work of Emmanuel Patin, who has been working on this now for the last year or two in the lab and has really made some very interesting observations, which I can share with you. So Manu's approach really has been to use in vivo analysis, looking at tumor microenvironment and immune profiling, identifying cellular targets for potential immunotherapeutic combinations. And I'm going to show you some of his data. So Manu's model is again in an immunocompetent C57 Black 6 mouse using the MOC2 head and neck system, so a syngeneic head and neck system, and again using radiation plus ATR inhibition. Now note the radiation dose is deliberately selected to be subtherapeutic at 8 gray in 4 fractions. And Manu has done a number of studies where he has been able to pick apart the immune landscape within the tumor based largely on flow. So the first data I show you is that in contrast to all of the intent on using, for instance, T-cell mediated responses to radiation, Manu's data point in this model to NK cells being a potentially useful therapeutic avenue. And you can see here on quantification of NK cells that there is a significant uplift in combination with ATR and radiation. Indeed, the ATR inhibitor alone and radiation alone increased the proportion of NK cells within the tumors. Those NK cells are displaying activation markers such as CD69, proliferation markers Ki67, and markers demonstrating functional activity, NKG2D, for instance. Importantly, they also show co-stimulatory markers such as 41BB and ICOS, potentially pointing to the way to combination therapeutic approaches. Those NK cells that we isolate are more mature, so you can show that there are CD11B high and CD27 low cells here with the ATR plus radiation combination. So these NK cells are not just more numerous and more activated and more proliferative, they're showing maturation, and they're also showing effector mechanisms in that they secrete more gamma, and they are also showing more evidence of degranulation based on CD107A. And you can see that the double positives for both gamma and CD107 are increased in the ATR plus radiation population, again indicative of a more active phenotype. Now, when Manu looks to see whether or not he can undo the therapeutic combination, so the green curve showing ATR inhibition with radiation with an isotype antibody versus a depleting NK 1.1 antibody, and on the right-hand side of the slide, you can see that he effectively depletes NK cells from the tumors and the lymph nodes. You can see that there is a partial abrogation of the therapeutic effect, not a total abrogation. Now, within those NK cells, he has been particularly interested in looking at what other markers there may be, and I've already shown you some data on ICOS and 41BB, but I want to just show you some data relating to his work with TIGIT. TIGIT positive NK cells are increased with the ATR plus radiation combination. And if we take this into a therapeutic model where we combine TIGIT with anti-PD-1 in combination with the radiation and the ATR inhibitor, again with the same subtherapeutic dose of radiation, what we can see is that we see an improvement in tumor growth delay, but notice we do not see cure. This is a particularly difficult model to cure, and Manu continues to wrestle with this and is assessing other combination regimens to see whether or not we can crack this particular issue. But we can see that there are modifications that we can make potentially to NK and indeed, of course, T cells, because TIGIT can be expressed on T cells, that can lead to improved therapeutic efficacy. So in the last couple of minutes of the talk, I'd like to return to the work of Magnus Dillon that is based around the Patriot study. So this is a phase 1 study that we've been conducting now for the last few years with an ATR inhibitor, both as monotherapy. You can see in part A, we did a dose ascending design. In part B, we did expansion cohorts based upon markers of replication stress, DNA damage, response deficiency, to choose patients who may be sensitive to these drugs as single agent. And you can see also that we've tested different schedules, continuous dosing and intermittent. Again, this work is the work of Magnus Dillon, and this was presented most recently at the ESMO meeting in Barcelona last year at a time when we could go to meetings. And you can see here, these are data from single agent responses. The black diamonds indicate the intermittent schedule 160 BD, which is our recommended phase 2 dose of this drug. And you can see that we have seen single agent responses in the axilla, in esophageal cancer, in the neck in head and neck cancer, and in the nasopharynx in undifferentiated carcinoma nasopharyngeal type, amongst other responders. But more interesting to this group, I think, is the combination with radiation. It has more relevance to what I've told you earlier in the talk. And so we have run a study with parallel cohorts with disease above the diaphragm and below the diaphragm, with initially ascending doses of drug with 20 gray in 10 fractions, then ascending numbers of fractions. So initially half the dose of radiation sensitized with the drug, and then the full dose of radiation sensitized with the drug. And again, we've been looking at this with a run-in of the drug sensitization throughout the course of radiation, and then 2 days of treatment thereafter with monitoring. And I show you some of the responses that Magnus has documented. So again, this is in the pelvis, a metastatic deposit around the acetabulum, showing a radiological healing of that. Again, a metastatic head and neck cancer responding in the mediastinum. And to the left-hand side of the slide, I show you the growth trajectories of irradiated tumors in the solid lines and the unirradiated tumors in the same patient, same color coding, indicating that in the patients where we didn't irradiate the tumors were growing relatively quickly, but when we gave the drug radiation combination, we often achieved profound responses up to and including what was looking like a complete remission. And in the level of anecdote, I show you 2 patients, both of whom presented with locally advanced head and neck cancer, judged to be inoperable and not fit for radical dose radiation, who had not previously been treated, who received either 30 gray in 15 with the drug or 20 gray in 10 with the drug. And this patient who received 20 gray in 10 is now at 3 years in complete remission after only 20 gray of radiation. The patient who received 30 gray in 15 is now at 12 months in complete remission after 30 gray plus the drug. So anecdotes suggesting there's sensitivity, and AstraZeneca is currently setting up a randomized phase 2 study in patients who are not able to receive platinum to receive full dose radiation. But there have been a number of translational components of the study that Magnus is leading on, including pre and post-treatment tumor biopsies, and also paired blood and paired plasma. And there will be, I hope, multiple interesting observations from that. Unfortunately, I don't have time to share the preliminary data with you, but we can perhaps touch on them in Q&A. So in conclusion, I hope I've shown you that this new drug, which now has a name, ceralasertib, is well tolerated. It has monotherapy effect. In preclinical data, I've shown you it's associated with mitotic death, with micronucleation. We've seen that in the clinic, it has, and in the preclinical studies, excellent responses in combination with radiation therapy, suggestions that some of this may be at least in part due to nucleic acid sensing pathways. And I haven't had time to show you some of the other more detailed data we have, but we have preliminary data that this is mediating immune modulation within the clinic as well as within the preclinical studies. So I close with a picture of the lab. I recognize Magnus Dillon, and if you can see my arrow, you can see him in the middle of the slide. Martin McLaughlin, who has really been absolutely central to this work within the team. Other members of the team listed there. I recognize the support of Alan Melcher, with whom I share a laboratory. And I also recognize the support of AstraZeneca and collaborating centers, notably Martin Forster at UCL and James Spicer at Guy's St. Thomas's, who've been really important in getting the Patriot study done. And most of all, of course, I recognize the patients and their families for taking part in these studies and contributing biopsies. Thanks, Tim, and others.
Thank you very much, Kevin. Really fascinating data on the ATR inhibitor. My first question is, have you compared any of the other DNA damage repair inhibitors, and have you reason to think there might be any differences?
Well, it's interesting you should say that. So Magnus Dillon, who is establishing his own group in his own laboratory, has been doing precisely that, and has been linking in and collaborating with AstraZeneca. Of course, AstraZeneca has a deep and very interesting pipeline of DDR inhibitors. So Magnus has been profiling the DNA PK inhibitor, the ATR inhibitor, CHEK1 inhibition. He's been looking at PARP and WEE1 kinase, and the ATM inhibitor, I mustn't forget that. And we imagined there might be differences, and indeed there are very striking differences across models. I can't share those data with you, first of all, because I can't remember all the details, and secondly, they're Magnus's data.
Yes. Okay, thank you, Kevin. I don't know whether you're able to look at the questions in the chat, but there are a couple of questions that have come in. One of them is regarding the PD-L1 expression that you see with the ATR activation. And the question is, do you generally see higher PD-L1 levels after AZD6738?
So in the animal models, we certainly see that. Interestingly, it appears to be quite profoundly on the non-tumor cell compartment, as well as some upregulation on the tumor cell compartment, as I showed you. We haven't systematically got the data from the Patriot study to be able to answer that question from the clinic, but the hypothesis is that we will see similar things, and that I might reasonably expect that that will be present with the drug alone, and maybe more profoundly present with the drug plus radiation. And then it says, have you tried to treat cells with DNA replication stress-inducing agents such as low doses, and then treat them with ATR and RT? We haven't done that experiment, actually. That's a complex combinatorial experiment. I think it's a really good idea, but it's not something that we've addressed at the present time.
Thank you very much, Kevin. In the interest of time, we'll move on to the next presentation from Adel Samson in Leeds, who's going to be talking to us about viral radioimmunotherapy for brain tumours. So, Adel, if you could share your screen, that would be great.
Well, thank you very much for the kind invitation for me to speak. So, in fact, I'm speaking on behalf of my colleague, Susan Short, with whom I co-lead the radiotherapy drug therapy theme within the Leeds RadNet Centre. So the data that I'm going to show you was performed with Susan, as well as with good colleagues at the ICR, Alan, Melcher, Kevin, and others. The theme of this presentation is on GBM, and I'll just give you a little introduction to it. So glioblastoma multiforme is a malignant grade 4 tumour diagnosed in over 200,000 people globally each year. Complete surgical resection is often difficult as tumour overlaps with normal brain tissue, and the median overall survival with standard of care surgery and temozolomide radiotherapy is only 15 months. Since 2005, we've had only 3 new treatments approved for the treatment of GBM: temozolomide, radiotherapy fields, and bevacizumab, which is used less in Europe and more in North America. So immunotherapy for GBM is our current interest, and there are various indications that immunotherapy could be successful in the treatment of this disease. One of those indications is that T-cell exhaustion is particularly prevalent in patients with GBM, with multiple upregulation of immune checkpoint co-inhibitory receptors on circulating T-cells, including PD-1, PD-L1, LAG3, and TIM3. However, there is a potential problem in getting the antibodies to the brain tumor, and that's the blood-brain barrier, which excludes many chemotherapy drugs and larger molecules such as antibodies. So in order for an immune checkpoint inhibitor to potentially be successful in the treatment of GBM, it must interact with immune cells outside of the brain, with subsequent trafficking of those immune cells to the brain. So immune checkpoint inhibition is now standard of care in the majority of solid malignancies. However, in GBM, it has failed so far. One of those important studies was a phase 3 study, the Checkmate 143 study, in patients with recurrent GBM, where nivolumab was tested as a monotherapy in comparison to standard of care bevacizumab, standard of care in North America, without any survival advantage in the nivolumab arm. Some questions were raised as to why that may be the case. Could it be a lack of tumor infiltrating lymphocytes? And these patients had received prior chemotherapy with temozolomide, which could have depleted their lymphocyte pool. Could it be poor antigen presentation and poor priming of the cytotoxic T cells? And that then brings up the question of the timing of immune checkpoint inhibition, potentially pre-surgery or when there is a significant tumor load in order to improve T cell priming. It also indicates a role for combination therapy, combinations with therapies that are able to improve T-cell infiltration into the tumor, to improve antigen presentation, and to improve priming of cytotoxic T-cells. And those potential therapies include radiotherapy, most obviously, as well as oncolytic viruses. So we know that radiotherapy is capable of stimulating immunogenic cell death. And that's a process that has been talked about during today's meeting. However, we also know that radiotherapy is capable of inducing non-immunogenic cell death. And we don't know which of these applies in the case of the current STUP regime of radiotherapy in GBM. What we hope is that radiotherapy is able to induce the release of DAMPs, leading to interferon and inflammatory cytokine secretion, which then results in chemokinesis and TILs, NK activation of the innate immune system, DC antigen presentation and maturation, CTL priming, MHC upregulation, and thereby systemic immunity, and also upregulation of immune checkpoint receptors and ligands that can subsequently be targeted by inhibitor antibodies. But we don't know if that truly does apply in the case of GBM. This potential theory was tested in two recent clinical trials, two large randomized clinical trials in GBM, whereby in the Checkmate 498 study, newly diagnosed patients with GBM were treated with either nivolumab or temozolomide in combination with radiotherapy. And unfortunately, there was no overall survival advantage in the trial arm. And then subsequently in Checkmate 548, again in patients with newly diagnosed GBM, where nivolumab was added to standard of care temozolomide radiation therapy. And again, there was no overall survival advantage. So two big trials that we had been eagerly anticipating, both showing that the addition of PD-1 inhibition does not improve therapy for these patients. And really that leads us to question whether current radiotherapy schedules are inducing non-immunogenic cell death in GBM, and whether we might need to give that process a little helping hand, potentially in the form of oncolytic viruses. So oncolytic viruses are therapeutic replicating viruses. They can be either engineered or wild type. They can be engineered to express transgenes, and they preferentially replicate in malignant cells, inducing immunogenic cell death. That leads to interferon secretion, the release of DAMPs, and the subsequent immune activation, stimulation, and potential abscopal effects that we've spoken about, and they turn cold tumors to hot. There are numerous ongoing oncolytic virus trials in combination with immune checkpoint inhibitors, which I won't detail. And the current lead agent in patients with GBM is DNX2401 and DNX2440. So these are engineered adenoviruses that selectively replicate in RB deficient cells. One of the agents expresses OX40 ligand, but these agents are only being given by intratumoral injection because of fears that intravenous injection won't result in the accumulation of virus in the brain tumor. So a number of other viruses have also been tested in GBM, all of them by intratumoral injection, and that's due to fears that the blood-brain barrier will exclude those viruses. However, this process requires neurosurgery, and it's obviously difficult to repeat neurosurgery in patients if you want to give a schedule of oncolytic virus therapies. The picture I'm about to show you is a bit gory. I apologize for that, but essentially that's what intratumoral injection in patients with GBM looks like. It's not a process that you can repeat on a full weekly basis, as you would want to do with an immune checkpoint inhibitor plus virus combination. So we set out to explore whether intravenous delivery of a virus can result in accumulation of virus in the tumor. And that's because intravenous delivery allows recurrent administration, fits into existing chemotherapy services, it's more cost effective, it theoretically allows widespread virus delivery into a tumor that's infiltrative, less invasive, more acceptable to patients, there's rapid recovery from the procedure, but there's a question as to whether we can get enough virus to the tumor and what happens in that tumor once it reaches it. So we set up a 9-patient neoadjuvant study of intravenous injection of a virus ahead of debulking of recurrent GBM, other high-grade tumors, or brain metastases. So this was a single center study. We treated 9 patients in total. The virus that we used was reovirus, which is a wild type virus. And the secondary objective after the primary objective, which is to see whether the virus reaches the tumor, was to assess the brain tumor microenvironment and whether that enables combination therapies in future clinical trials. So the results of that study showed that reovirus does indeed reach GBM successfully, and we showed that in a number of different ways: by immunohistochemistry, RNA in situ hybridization for the viral genome, by electron microscopy immunogold, QRT-PCR, as well as immunofluorescence. So the theory that the blood-brain barrier excludes intravenously injected viruses from reaching the brain tumor is potentially wrong. And then looking at the brain tumor microenvironment, we found that there were changes in peripheral blood T cells in terms of upregulation of ICAM, so intercellular adhesional molecule, which indicates that those T cells are moving from the blood vessel compartment into the tissues. They're extravasating. And that's a process that we could see actively in one of the resected brain tumors, whereby these CD3 labeled T cells in brown can be seen infiltrating through the blood vessel wall and into the surrounding tumor. And we found an increase in CD8 T cells into the tumor, so increased CD8 TILs following virus infusion in comparison to controls. We also found that intravenous virus infusion results in upregulation of PDL1, both on PBMCs as well as on TILs, virtually all of the TIL subsets, as well as on tumor cells. So that indicates that we are able to increase tumor infiltrating lymphocytes with a virus infusion and to increase PDL1 expression, which normally inhibits virus-mediated immune effects, but in the context of an immune checkpoint inhibitor could provide synergistic effects. Taking that work forward, we then wanted to see if we can set up an in vivo model testing that theory that virus infusion primes for subsequent PD-1 inhibition. Firstly, we tested whether the combination of reovirus with GM-CSF is superior to either agent alone. And that's data going back a long time with Alan Melcher and Richard Weil, which indicated that GM-CSF can increase the delivery of reovirus on immune cells, attached to immune cells, subsequently to the tumor, and that it can increase antigen presentation and therefore immune-mediated effects. And indeed, the combination of reovirus with GM-CSF was superior to either agent alone. And taking that a step forward, we took that combination GM-CSF with reovirus and combined it subsequently with a PD-1 inhibitor and showed that the combination produces synergistic effects in comparison to either agent alone. So really, that provided the evidence that we needed to take this further forward to see if an oncolytic virus can enhance standard of care chemoradiotherapy in patients with GBM. So just to summarize that section, IV injected reovirus reaches high-grade brain tumors, including GBM. There's an increase in TILs and upregulation of PDL1, and that produces synergy when combining a virus with anti-PD-1 therapy in an orthotopic syngeneic murine model of GBM. So taking that forward to see if GM-CSF plus virus can improve standard of care chemoradiation, we firstly tested this in an orthotopic syngeneic murine model, and we found that the combination does indeed improve upon chemoradiation alone, although not greatly, but there was a significant improvement nonetheless. And we then set up a phase 1/2A clinical trial in patients with newly diagnosed GBM, that's called the RioGlio trial, whereby we tested standard radiotherapy, that's the Stupp regime, given over 6 weeks, 60 gray in 30 fractions, alongside continuous temozolomide chemotherapy, followed by an adjuvant period of 6 months of temozolomide alone, in combination with GM-CSF and reovirus. Reolysin is the trade name for reovirus. Reovirus was given after GM-CSF over 2 cycles, initially with the chemoradiation and subsequently with each cycle of adjuvant temozolomide. The doses increased in the phase 1 element from 5 times 10 to the 9, 3 times 10 to the 10, and we successfully treated 20 patients in total. Now, the RioGlio study results, I'm afraid I'm not allowed to share with you at the moment, as they will be revealed at the Society for Neuro-Oncology annual meeting. There's an oral presentation which I'll point you towards. That's November 19th to 22nd this year. Future directions. So we're aiming for a clinical trial of standard chemoradiation with an immune checkpoint inhibitor, PD-1 inhibitor plus virus. That's ongoing work in Leeds with a new company, VCN, that produces an engineered adenovirus. And that's work, again, that's shared with Alan Melcher and other good colleagues. So the ideal trial that we'd like to set up is standard chemoradiation in patients with newly diagnosed GBM. In arm 2, that would be in combination with PD-1 inhibition and the virus. And in arm 3, we would look to drop the temozolomide, as there's a concern that that could be lymphodepleting and that would inhibit some of the immune-mediated effects. And we would go with radiotherapy, PD-1 inhibition, and virus alone. There's a second question about revisiting radiotherapy schedules, doses, fractionation in the age of immunotherapy, and whether the Stupp regime from 2005 is applicable to current modern day treatment regimes that we're trying to set up. So thank you very much for listening. This work involved a very large team of colleagues and collaborators. I'll take any questions, thank you.
Thank you, Adel. That was great, fascinating data. So the intravenous delivery of reovirus to GBM, it was surprising that you saw it in the brain. Is this secondary to interruption in the blood-brain barrier, or might this be seen in normal brain and we just basically the dogma was all wrong?
Yeah. I guess it's impossible to know whether that's the case or not in patients, as the surgeons are very careful not to take any normal brain tissue, as you might imagine, during surgery. These patients do have a disrupted blood-brain barrier. They tend to have edema, so angioedema, and that indicates that those blood vessels are leaking fluid. If they're leaking fluid and those patients need steroids to manage their pressure symptoms, then quite possibly virus is able to get through that disrupted blood-brain barrier. We don't know for sure. There's another element of virus hitchhiking onto immune cells, which is work done by Alan Melcher and Liz Eilert in Leeds at the ICR, that virus is not free in the plasma, that it binds to immune cells, and those immune cells are able to deliver the virus to tumors. We don't know if that applies to every virus, but it certainly applies to reovirus.
Okay, thank you. And your 3-arm trial seems rather ambitious. Would you have sufficient numbers in each of the arms to be able to draw any conclusions? It is an ambition at the moment, Tim, so it's not something that we currently have funded. GBM has been notoriously difficult for us to persuade oncolytic virus companies to do their research into. That's because they deem it to be a difficult tumor site, and these small biotechs are developing agents that they want to eventually sell off to pharmaceutical companies. So we've had to go back to step 1 initially,
with VCN, the engineered adenovirus company, just to see if that virus is able to reach brain tumors following intravenous infusion as the first step. So that big randomized trial is an ambition at the moment that we hope to reach in the future.
Okay, thank you. Are there any other questions for Adel? Could I ask you to stop sharing your screen, Adel? And it's a pleasure to welcome Awen Gallimore from Cardiff University, who's going to talk to us about new approaches for optimizing tumour-specific T-cell priming. So big welcome to you, Awen. Thank you so much for joining us today.
Fantastic. Thanks very much, Tim, for inviting me. And I assume I'm all set screen-wise, everyone can hear me and so on. Okay, so I'm interested in looking at approaches for optimizing tumour-specific T-cell priming, and in particular in finding out where exactly can we create this perfect storm. So what do we know so far about radiotherapy and T-cell priming? Well, we've heard a lot about this morning. We want to create inflammation to produce a source of antigens that can result in priming and a T-cell response that can then be sustained. And a lot of effort has gone into various steps here, such as trying to enhance priming of T-cells using agonist CD40 antibodies, as Tim talked about, also TLR7 antibodies, CTLA4 blockade, Treg depletion, and so on. It's also important to sustain the T-cell response, and here PD-1, PD-L1 blockade, and again Treg depletion has proven useful. So where does priming of T-cells then normally occur? So it normally occurs in the draining lymph node. And if you look here at a draining lymph node, the structures that I want you to pay particular attention to are these high endothelial venules. So these are blood vessels that are found only in secondary lymphoid organs, and they're found in the T-cell zones of the lymph node. And here they serve a very important function. Through virtue of the fact that they express peripheral node addressins, they can bind to the receptor L-selectin on naive T cells. So these are the blood vessels that enable naive T cells to access, to enter the lymph node from the blood. And because HEVs are absent in other tissues and organs, that is also therefore the reason why naive T cells don't get into those organs, and why priming is something that happens in the draining lymph node. We do sometimes see these structures at sites of chronic inflammation and autoimmune disease, and these are thought in those contexts be part of the pathology of the disease by allowing entry of T cells that essentially shouldn't be there. So what happens then normally is you have your tumour antigens expressed by dendritic cells will travel in the lymph to the lymph node, which may or may not have tumour cells in itself. And that's a site where these antigens then get to meet naive T cells. But we know quite a bit about, draining lymph nodes, immunotherapy, radiotherapy, and cancer in general. And so there's a few things that we have to think about for how we're going to induce the best T-cell response that we can. We know that there are several studies which show that the tumor draining lymph node is an essential entity. It's indispensable to successful immunotherapy. So it may be then that if your lymph node isn't functioning properly, such as has been described often in the case of sentinel lymph nodes, i.e. the tumor draining lymph node, then this may represent a serious impediment to successful priming of T cells. They've been described to have poorly functioning antigen presenting cells, even actually when tumor cells are not detectable within the draining lymph node. And the high endothelial venules, the HEVs, the blood vessels that you need to make sure these interactions take place properly are often found to lack the phenotype that is associated with a good anti-anything T-cell response. Then in the case of radiotherapy, there's even conflicting evidence, and Tim, you alluded to this morning, on the impact of irradiating draining lymph nodes on the potential for inducing good responses to radiotherapy. So with that in mind, I'm now going to talk about trying to do something slightly different, which is to try and develop some of these structures, these high endothelial venules in the tumour itself. So essentially meaning that we're trying to turn the tumour into a lymph node. And I'll tell you a little bit about what we found out about this in a carcinogen model of fibrosarcoma, where essentially what we do is we inject mice with methylcalanthine. Most of you have read the old immunosurveillance, or actually the continuing immunosurveillance literature, will know that this is the tumour of choice by the lab of Robert Schreiber. It's a carcinogen that we inject into the hind leg of the animal, and what happens is that fibroblasts will accumulate at the site of the carcinogen in an attempt to wall off the carcinogenic fibres. And these then acquire mutations and become eventually a fibrosarcoma. And what we find, as Sergio talked about, actually, this is a tumour that has many neoantigens because of the mutagenic nature of the carcinogen. We find quite a heavy infiltrate of T cells, and we certainly see a lot of tumour infiltrating regulatory T cells. So if you look at this graph here, we're looking at the proportions of Tregs in non-draining lymph nodes, draining lymph nodes, spleen, blood, and you can see that there's a highly significant enrichment of Tregs at the tumour site. So we asked the question, well, what happens when you take the Tregs away? We have done this using a genetic model whereby we can deplete Tregs using diphtheria toxin, as the FOXP3 positive cells express the diphtheria toxin receptor. And what we see is that when we compare tumour growth rates in our mice that have Tregs and those that don't have Tregs, is that we see a significant reduction in tumour growth rates in these Treg depleted animals. But we see some tumours that actually regress. And when we've compared these regressor tumours with the progressor tumours, the main difference that we see is in the level of T cell infiltrate. So here you have a tumour that's untreated, we have a non-responding tumour and a responding tumour. And we've stained them all at the same time with anti-CD3 antibodies. And you can clearly see here that the most obvious difference here between a responding tumour and a non-responding tumour is that these are heavily infiltrated with regulatory T cells. And for one reason or another, which I won't go into right now, we looked at the blood vessels in these tumours. And what we found was that in the responders that had high frequencies of T cells, we were seeing vessels that were staining positive for PNAD, so the marker of high endothelial venules, and also MASCAM1, which is a marker of immature high endothelial venules, immature HEVs. And they all stain with the pan-endothelial marker CD31. And what is absolutely clear, as I've already mentioned, is that when we see HEV positive tumours, these are the ones that are packed full of T cells, and they're the ones with the slowest growth rates. And we've done this work, of course, in the genetically engineered mouse model, but it's all been recapitulated using inhibitors of Tregs, such as PI3 kinase delta inhibitors. And there again, we see neogenesis of these high endothelial venules in some tumours, and this then corresponds with a increased frequency of tumour infiltrating lymphocytes. And so the key thing to consider here, well, if you have high endothelial venules in situ and you have a better anti-tumour T cell response, then it is quite likely that the priming is occurring in the tumour now itself, because by virtue of having the HEVs, naive T cells are now able to enter the tumour mass. So in order to try to understand a little bit more about these HEV networks in the tumour, to understand how extensive they are and so on, we've developed three-dimensional imaging techniques so that we can look not just at a slide and stain in a two-dimensional structure, but to gain some sort of overview of the entire tumour. So what we have done is we have injected mice with antibodies that are labelled and which can pick up an antibody called MECA79, which can pick up PNAD, so a marker of high endothelial venules, and somatolectin, which will stain all the vessels. The tumours and the lymph nodes are excised, fixed, bleached, cleared, and then you can look at the the whole thing using an ultramicroscope. And this is work that's been done in my lab by a PhD student called Stefan Milutinovic. And so the whole optimization has actually been done using lymph nodes. And I'll show you a little bit on that first. So this is a stained lymph node, stained with MECA79 and the tomatolectin. This particular lymph node that you're looking at is a tumor draining lymph node. And if you look at this slide here, you can see the differences, between an unchallenged lymph node. So this is just taken from a naive mouse to a sentinel lymph node. So this mouse has a tumor. This is the draining lymph node. And then you have on the other end an immune reactive lymph node. So this happens to be a lymph node from a Treg depleted mouse. And so we've used the lymph nodes to try to develop our computational tools, because it's actually quite a challenge to then first of all, generate these images, but then to have the computational tools that can enable you to measure the HEV networks. And you can see from some of the values that we've extracted from this that there are some quite interesting things to observe. So you're looking at lymph node volumes here. The sentinel lymph node hasn't gone up massively, but when you look at the length of the HEV network, that's expanded threefold. So the HEV network has now become extremely dense in this sentinel lymph node. In terms of the immune reactive lymph node, we see something quite different. Volume-wise, it's increased massively, but in terms of the total HEV length, it's actually about twice the size of the sentinel lymph node. So we're looking at features that correlate with immune reactivity or being a tumor draining lymph node that are quite different. The sentinel lymph node seems to be very tightly packed with HEVs, whereas the immune reactive lymph node is far less dense. And so far, we don't really understand what this means in terms of priming T-cell responses, in terms of, how well does that lymph node function? And if we were to, let's say, put these things together with conventional confocal microscopy or with two-photon microscopy, I think you could start to learn quite a lot about what defines the optimal functioning lymph nodes. And I think these are quite interesting techniques that maybe we could apply to looking at irradiated lymph nodes or the lymph nodes of irradiated tumors to see how they deviate from the norm. But anyway, the focus of this talk is on the tumor, and this is why we developed these tools, so that we could look at networks in the tumor. So the green is a high endothelial venule, and here we're looking at non-treated tumors. We're looking at non-responders, where we see almost no vessels, nothing that looks like a real vessel anyway. Then you have degrees of HEV networks within the responders. Here we see some, mainly around the outside of the tumor. And here we've got a responder where we're really seeing HEV networks throughout the entire structure. And what we know is that if we measure HEV lengths within a tumor, then the extent to which we have an HEV network correlates with how many T cells we have in there and how good a responder it really is. And I've shown you everything in 2D, and these are of course 3D images. So I've just added this slide so that you get a sense of what we are able to look at. This is one whole mouse tumor. So we also know the mechanism through which we can drive neogenesis of these HEV. We know that if you take the Tregs away, we activate T cells. These produce lymphotoxin alpha-3 and TNF-alpha, both of which are cytokines that are essential for driving development of these vessels in tumors. So what we think we're setting up here is a sort of self-amplifying loop whereby we lower the threshold for immune activation, in this case by depleting Tregs. We're activating T cells. These are then helping drive development of these vessels in the tumor, which allow in entry of naive T cells. And so the self-amplifying loop continues, resulting in tumor destruction. And other labs have shown the same principle using slightly different methods. So the lab of Victor Engelhardt has shown that if you adoptively transfer enough activated T cells, then the same thing happens. And in the context of these experiments, because they are transferring in their T cells, they can actually definitively show that naive T cells are primed within the HEV positive tumor mass. And then the labs of Gabrielle Burgers and Ruth Gans have together and separately generated actually a body of evidence that you can drive HEV development in tumours, not just by lowering the immune threshold, but you can help this along by adding in blood vessel normalizing agents such as anti-VEGF. So if you normalize your blood vessels, this facilitates the process of HEV development with an immune stimulating agent, such as in their case, anti-PD-L1. Now, we also know in the case of radiotherapy, well, at least it's in the literature, that you can drive activation of dendritic cells in situ through radiotherapy. So those dendritic cells could prove extremely useful for priming T-cell responses in situ. So what else then do we know about tumour blood vessels and radiotherapy? Well, I only know what I read about, so hopefully it's all true, that well-oxygenated cancer cells are more sensitive to the cytocidal effects of radiotherapy than hypoxic cells. And these sorts of observations, as I understand it, have led to early phase trials which attempt to combine inhibitors of the VEGF pathway, that's to promote vascular normalization, with radiotherapy. So we know that hypoxia also negatively affects T-cell function. Hypoxia also promotes immune suppressive pathways, such as the accumulation of myeloid-derived suppressor cells and even anti-regs. So vascular normalization also improves immunotherapy. So this is actually now my last slide, where, of course, you'll have found me out by now as a radiotherapy imposter, as I've gone through all my data slides, and clearly I have not done a single radiotherapy experiment. So perhaps, it's a bit much to end with, having also learned about the complexities of finding the optimal radiotherapy schedule with the right dose, etc., to suggest that this could be a three-way approach, incorporating immunotherapy, radiotherapy, and also anti-angiogenic therapy. This has, as a triad, been suggested by others as a way to increase the perfusion of the tumour to enhance the cytotoxicity from the radiation, and also to increase effector T-cell trafficking, because vascular normalization agents are known to increase VCAM1, ICAM1, CXCL9, CXCL10 expression, all of which serve to facilitate infiltration by effector T cells. But in addition, it might also be reasonable to suggest that such a three-way approach might also promote development of high endothelial venules to enable in situ priming of naive T cells, which may be particularly important in the case of radiotherapy, given the complexities of whether you should or should not irradiate a draining lymph node and so on. So I shall stop there by mentioning the key players of the team. Emily and Kohlberg and James Hindley developed the HEV, the methylclanthurine HEV model, worked on that for some time, whereas Stefan has developed all the approaches for the three-dimensional imaging. And our indispensable collaborators have been Jens and June at Freiburg, and also Gib Bogle, who's actually from Auckland University and who's been extremely good at helping us develop computational tools for tracking the high endothelial venue. So thank you very much.
Well, that was great, Awen. Thank you indeed. Amazing images. So when you get the expansion of those reactive lymph nodes and that self-amplifying loop, do you have any idea of what controls the cessation of that self-amplifying loop and the process of regression of the lymph node?
Well, in the case of a pathogen where this has been, studied in far more controlled situations, then it's the disappearance of antigen that controls the retraction of the immune response. So as the antigen it leaves, so it's controlled as part of the immune response, then the immune response dies out, and then the lymph node a little bit later will shrink back to its, not quite its original size, but pretty much so. Yes. So antigen is what controls it under, sort of normal immune conditions.
Owen, can I direct you to the chat to see the questions that are coming in? There's a question from Kevin Harrington about the presence of HEVs in tumours associated with tertiary lymphoid structures in vivo and all the B-cell infiltrates is the presence of HEV in tumours associated with tertiary lymphoid structures in vivo and or B-cell infiltrates?
Yes, so you can, first off, you don't have to have a tertiary lymphoid structure in order to see the enhanced infiltration by T cells. So in the case of the tumours that I've shown you now, we rarely see TLOs actually. So you'll see the high endothelial venules and they'll be surrounded by scattered T cells. They'll certainly be at a higher density around the HEVs than they are at other sites in the tumours, but they won't be a particularly sort of well-organised structure. Yet, from everything we can see, they are participating, you know, in the anti-tumour immune response, because if you deplete the T cells, then you lose the effect. But I think that's actually the fact that we don't see TLOs is really because we're only observing these tumours for a few weeks. And I think if you allowed the whole thing to, you know, sort of carry on, eventually you'd end up with TLOs. And in fact, with Gabrielle Berger's and Ruth Gantz's models, where they use the blood vessel normalizers, then there, I think you drive the TLOs a little bit more quickly. And you have to have an HEV to have a TLO because they are, you know, an organizing component of the entire structure.
Thank you. So there's a question about anti-CTLA-4 with ipilimumab. Have you looked at these ipi-treated patients and whether they develop HEVs in their lesions?
Yes, so I haven't, but there are, there were a couple of papers actually this year, I think 2020, you know, it's been very easy recently to completely lose track of time and the month and the year and so on, but I'm pretty sure it was the beginning of the year. There were a few, there were a little spate of papers in Nature looking at TLOs in tumours of patients treated with anti-PD-L1 with or without anti-CTLA-4. And yes, they do show that you do see these TLOs there, and in fact that they are associated with a positive response to therapy.
Okay. A question, I think, from Richard Adams. Do the neoantigens themselves drive the HEVs? I.e., can we link the MSI high tumours with HEVs and the mechanism of infiltration?
Yes, well, probably. So this is a question we don't know the answer to. So back to my, you know, sort of very first 2 or 3 slides where I showed that only some tumours respond, you know, and it's the responders develop HEVs, and we hypothesize that those are the ones with the strongest neoantigens that can get a good immune response off the ground in the first place. So it's a hypothesis, but yeah, I would have thought the whole thing is linked, you know, neoantigens, immune activation, generation of these structures.
Do you have any evidence of perfusion of the HEVs in the tumour from Divine Mukherjee?
We have not measured perfusion in these tumours.
Okay. Are there any other questions for Awen or others? Okay. So what I'd like to do in the remaining time, and I won't keep us long, but I did particularly want to discuss the way forward as to whether there was any enthusiasm from the speakers and participants this morning on forming a RadNet working group for radiotherapy immuno-oncology with the preclinical models and sharing of expertise with clinical trial development and collaboration. So there is a networking fund associated with the RadNet Awards that enables us to potentially fund pilot projects. So firstly, any comments or thoughts about a RadNet working group in this space?
Tim, it's Alan here. I think yes is the short answer. I think there's enough interest and shared expertise, particularly across the preclinical and clinical interface, to make that a worthwhile enterprise. I'd be supportive.
Any other comments?
Tim, is Kevin. I second the motion.
And I'm also very much for it. It's Adel Samson.
Thank you, Adel.
It's Anna Wilkins jumping in. I think sharing of preclinical models for someone at my stage trying to be a junior clinician scientist would be particularly helpful.
Yeah, it's good. Thank you. Nice to hear you, Anna.
Hey, Tim, it's Amato. I like to also think this is a great idea, and I think there's a lot of very exciting things going on. And I'd like to thank you for organizing all of this today.
Pleasure. It's been fun to have such a quality speakers this morning. A real highlight in a challenging year for me, at least.
Yeah, I would also like to second that. Thanks, Tim, for a great meeting, and I'm all in favor for this working group.
Who was speaking? Sorry,
Eric Vanderberg. Sorry, I recently joined as a team leader at Radiotherapy in Imaging. I was in Sandra De Maria's group earlier.
Oh, great! Oh, great! Oh, nice to hear you, Eric.
Yeah,
yes,
yeah. Sorry.
Yeah.
Introduced myself.
No, very nice to hear your voice. Okay, I don't know whether we've got anybody from the NCRI CT RAD on the line, but the other...
me, Tim, that's helpful. Richard.
Oh, Richard, great. Thank you very much, Richard. So I wondered whether you'd like to say anything about the development of a meeting next year, in particular with the British Society of Immunology, and how we might input into that, particularly regarding clinical trial development with radiotherapy and novel immunomodulatory agents, so we can put our expertise together to develop the best trials, and also the interesting area which I hope we can involve cardio-immunologists in with these the radiation-induced immunological mechanisms of normal tissue injury in the gut and the lung. So, Richard, what are your thoughts as regards the CT RAD as to how we might integrate that into a national meeting?
That's very kind of you, Tim. I mean, I think this would be, the platform, first thank you for this meeting, has been phenomenal, as you say, great speakers, great science, great to share that. And I think that's one of the things, I suppose, through NCRI, we try to take further to the next step. So CT RAD has been focused, particularly in the group that I chair, around the development of early phase clinical trials, particularly with agents in combination with radiotherapy. And that's a great stepping stone forward from the work that you're demonstrating here. So we've previously run meetings which have been a mixture of preclinical scientists, clinicians, and obviously key to that, methodologists from trials units around the UK, to really try and take these to the next step, to look at the outputs of the preclinical data, translational work, and how we move that into a clinical trial arena, but also to look at some of the early phase 1B work, and actually see how that should be then designed and developed into phase 2 and phase 3 trials. So CT RAD, it has that remit. It's a UK-wide remit, and hopefully therefore is very inclusive. And we'd be very keen to work with people in the future, as we have done in the past. And Tim, you've been a fantastic advocate and previous leader of CT RAD. But Kevin, Alan, a number of people on this call have been very engaged in CT RAD in the past. And stepping forward with RadNet to try and help deliver those research concepts into the clinical forum would be great. What we're trying to do is working with, again, very much with BSI, is develop a meeting probably in the sort of late spring next year. Again, we're looking at potential virtual platforms in preparation for that. But it would be great to talk with you and other members who are on today in terms of how we develop that further and meet the needs of the community, really.
Thank you, Rich. So it seems to me there are two...
Tim.
So go on.
There's Mark Coles. I happen to be the BSI Congress Secretary coming in. So sit on the committee, and I'm certainly taking this on board, and I will make sure that this is brought up in the next meeting. I'll discuss it with Doug as well.
Thank you, Mark. So, Richard, it seems to me that there are two potentially quite different areas, complementary but different in terms of clinical trial development, and then the other more mechanistic data that is maybe with the translational science and the preclinical science. What are your thoughts about how we might best integrate with the BSI?
Yeah, I think that's a one of those nub questions, isn't it? I think we've been challenged more broadly across the UK community and specifically by the NCRI and their new strategy to make sure that we get better engagement between preclinical scientists and clinicians and everything in between. And I think the best format for that is still something I think we need to play with. And I think the structure of the organising committee for that next meeting should have the voices from those different groups. Valued thoughts from this group. I mean, how do people feel that we best integrate? There are people, as Tim, you're a great example of this, who sit at that, straddle those groups very effectively in your working with the labs and with delivering on a clinical front. And Kevin, again, very similar there, and Alan also. So again, advice on that is greatly received. Do you do 2 different meetings, or do you do some form of hybrid? I suppose I have some of my own thoughts around that, but I'd value anyone else's input, really.
Could I ask Kevin or Alan to comment, and then others, please? Would anybody like to comment on the format of the meeting, please?
Well, comment on what the format of this meeting or the meeting?
No. Thanks, Amato, for coming on. This is a meeting in March with the NCRI Clinical Translational Radiotherapy Group, interactions with the BSI. What are your thoughts about how we might mix 2 potentially separate but complementary agendas of clinical trial development and mechanistic science?
Sorry, Tim, I couldn't call in earlier. Alan here. So I think, I mean, it's about the overlap and interaction that I think is really the focus. You can't do everything, and it's only an opinion, but maybe particularly to pick out the translational interface. So far be it from me to say that, you know, our rectal cancer data, from what I've heard from you, plans to do similar prospective tissue collection.
Yes.
Is that an area to focus on rather than covering right through from mouse data through to therapeutic trials? Just a thought.
Yes, and thank you for that comment, Alan. But how would you see the interactions with the BSI working best in that then? How do we really get immunological mechanistic science pervading what we do in the clinic?
Yeah, well, I suppose what again, perhaps we share an interest in trying to understand the basic mechanism science from the human analysis that we do. And as you know, you and I, and with Manchester and Anna and others, we have an example of a clinical trial in bladder cancer called REARM, which is funded to look at some of these translational analysis. The honest truth at the moment is this sort of stuff is more aspirational than real data. So I take the point, and I'm not sure what the answer is, but perhaps to pull in both more clinical and basic science, I think there is an opportunity to look at mechanistic data from human rather than mouse alone data.
So are you saying, Alan, that we should have everybody in the same room, or 2 parallel streams?
Okay, help, don't know, on the spot.
Any comments?
Any others?
Tim, sorry I couldn't comment. I was on the phone to somebody else and I just heard your things out of one ear, really. I guess that I would endorse what Alan says about exploiting the expertise of those who know, you know, a great deal about immunology that we as cancer immunologists may not necessarily focus on a day-to-day basis.
Yeah.
And so I think that would be a really nice opportunity to get some of the basic science embedded within some radiation related and cancer related topics. I guess the other thing that I would perhaps highlight is, of course, I think some of the immunologists may spend quite a lot of their time trying to shut down immune responses, whereas of course we spend, I think, or we're beginning to spend our time trying to activate immune responses. And there may be occasions where shutting stuff down may be important, not least of all in relation to mitigating toxicity. So I think some interface there would be really useful to learn from immunology about some of the suppressive as well as the activating approaches. And in terms of how to do it, I think that's going to be difficult, isn't it? Because it's 2 groups of people probably speaking slightly different languages at the beginning. And so I would imagine mixed workshops and a mixture of speakers from both sides trying to find common language and common areas of overlap.
Thank you, Kevin.
So this is Mark, just quickly, if you go further into the future, say 2022, you know, often we do a joint Congress session with PIN, for example, with immunodeficiencies. It might be possible, with longer discussion with Doug, and by then I'll be Congress Secretary, to have like an NCRI BSI kind of joint meeting. but that would obviously be 2022 would be the earliest that could be discussed. But if we want to have a bigger sort of meeting, I mean, even the Congress meeting we're having, the virtual Congress meeting in December, there will be a cancer immunology session because we know it's very popular with the participants at BSI Congress, and you'll probably hear about that shortly.
So I think that, Mark, would be a great thing to work to, particularly if you might be in a strong steering position by 2022. And the other attraction to such a meeting is we've got plenty of time to organise it, and there is actually a sporting chance that we might be able to see the whites of each other's eyes.
Tim, it's Awen. I just wanted to mention that the BSI have a tumour immunology affinity group. Yeah. And, you know, that's run now by a group of people. Ben Wilcox is the chair, I think, of it. So he would be a very good sort of person to interface with to create the, you know, the ideal meeting for this, perhaps. And that tumour immunology group was, you know, reinstated actually in the last sort of 18 months by BSI.
Okay. So, any additional thoughts, Awen, from your perspective as to how we might best interact and build this?
I think the NCRI BSI groupings already occurred actually, so that has had 1 or 2 meetings already, and it's decided, I think, that its focus is going to be, well, at least initially, on the adverse events following immunotherapy. So I think there's a couple of people on here actually who are part of that group, so it might be worth discussing that by Mark and Doug already. Okay. So that entity already does exist, that structure. Okay. I on the line, and she's part of that too.
Okay.
Can I make a comment, Tim? Is Eileen Parks here? Is Eileen Parks here from Oxford?
Thank you. Please go ahead.
Would you think of something like a sandpit for this? Because having a meeting where we discuss potential combinations would be really helpful, but maybe something like a sandpit where the goal of the meeting is to get collaborations developed where we have this pilot funding awarded at the end of the meeting, the way that the CRUK sandpits are run.
Yeah, I think that would be good. Any other comments from anybody else?
I'm certainly, Tim, I'll come back in because I've been listening to people's thoughts, because that's what I wanted to do, I suppose, from my point of view, is that they're all very, very useful input and thoughts. I think obviously the concept of the CT RAD BSI one very much does have a radiotherapy focus, and maybe that can be used as a developmental component of working with the BSI. As Awen's already indicated, there is the cancer group with working with NCRI and BSI, which I think is very useful. Come back to Eileen's point, and the previous way we've run these is to have an element of sandpit in relation to it as well, so that's well received. We've often had people present fairly simple ideas which they submitted to the group in advance to say, look, I've got this idea of how we might take a trial forward, and I'd like to ask questions from the group around, you know, what translational work we should do, what preclinical additional work we might need prior to actually persuading pharma to engage, and then how do we actually deliver their trial and in what tumor types, etc. So people come with concepts which can be quite well developed in certain circumstances, or actually quite early stage, at which there is that sandpit opportunity really to actually get greater engagement and collaboration. So I think that would be something that the group would be keen to maintain, and maybe greatest opportunity to get engagement across the fraternity of preclinical science through to clinical science as well.
Thank you, Richard. Are there any other views or comments to make from colleagues? Well, it just leaves me really to try and briefly summarise. I think we've had a really excellent morning, and I'm extremely grateful for all of the speakers putting in the time and effort to present such interesting and diverse data this morning, and for you, the audience, in making it such a worthwhile experience this morning. We've got a lot to build on. I think there's an awful lot of really good work that we can potentially do together to make us internationally competitive in this area. And I think the next big opportunity as a building block will be the meeting that we're talking about with the NCRI, CT-RAD and BSI, but also to work within the RadNet network as developing a working group, working closely with the partners as we've discussed. So thank you all very, very much for joining this morning, and we will follow up and start to build another meeting into next year. And please don't hesitate to contact any of the speakers that have been online to start, if you've been interested in particular areas this morning, to start to build those collaborations across the network. Thank you all very, very much.